All right. Yeah. So now it should actually be going in reality.
Let me go on.
No, in real life.
Let's
Not online, in real life.
Kyle and
Yeah, I think it's happening. I think it's true. Okay. Man, yeah, there was a big OBS update like in the time that I have not been using OBS, and now it just has this new thing called broadcast management, which is just like you have to click 2 more times for absolutely no reason. Anyway,
yeah, it seems like it's on. It says on your thing.
yeah, I think we have 1 concurrent viewer, so that's pretty cool.
That's probably
Yeah, that's it. Okay, welcome everybody to Kyle and Friends, episode 12, with Thucydides, and we're talking about really exciting stuff, null-hypothesis significance testing. Yeah, so, somebody said my mic is making weird vibrations. How about now? Like,
let me go on the channel. See if
yeah. And it's I just love this. I love the streaming technology. Like,
let me go on the channel. Yeah, you do have like a tiny reverberation on your mic.
yeah.
Like zzz, it's a weird thing.
It's still.
Let's see. Let's go back. And yeah,
It's still.
do you hear it? Let's see. Let's go back.
No, I do.
And yeah.
Yeah.
What if you use without the mic?
Yeah, I'm gonna try changing it. Oh, wait a second.
Let's see. Let's put it back.
Still. How about
Do you hear it? Let's see. Let's go back.
No, I do.
Let me reload really quick so it's up to date. Let's put it back. Still. Do you hear it? Let's see. Let's go back.
No, I do.
Let me reload really quick so it's up to date. Yeah. Let's put it back. Still. Do you hear it? Let's see. Let's go. Could it be possible that because I'm not using a speaker, it's like getting a mixed input? I mean, a microphone. I'm using the computer speaker.
Okay.
Same thing. No headphones. It's just purely coming out of the laptop. Let me try. Let me just try putting headphones on, see if it helps.
I don't think so. What are you using to listen? That could be a problem, but I would feel like it would not. It would have an echo.
Let's see. Can you hear me fine? Let's put this thing on.
Yeah. Yes, I can. And
Yeah. Trying to. Oh wait, I think you're. Unless that's me now, I can't tell anymore.
Not great. Not a great situation.
No, I'm just streaming. No,
What
it's
are you
still
listening?
you still your headphones or microphone in this case. Mine sounds completely okay in the stream.
Yeah. You sound okay to me through Skype, but yeah, I can't tell from the stream. I should be monitoring the stream, but
Take back, reload.
all right. How about this? This should
Take
be
back,
different.
reload.
It should be
All right, so
all right.
the computer's not picking up your sound now. I think you have to switch
How
from
about
microphone
how
input
about sound effects?
to the computer input on audio settings. So I can't hear you anymore, just so you know.
How about now?
That's great. Let's see if it works on the stream.
So yeah, I think I turned off.
How about that's awesome. It works perfect. That's great. Let's see if it works on the stream.
Oh, really?
Yeah, I think I turned off.
My God.
That's awesome. We're good to go, I think.
Well, it wouldn't be. I mean, this is just par for the course, right?
Podcast.
Yeah, I don't know why. Usually I just do NDI for both people, but it was like, yeah, wow, okay. All right, so anyway, null hypothesis significance testing. So AKA, P sorry, let's just
Sure, man. Whatever you want to do.
let's just like let's just chill. So so I haven't streamed for a pretty long. We'll just call this season 2 of Nutrichronology, Kyle and Friends, etc. Season 1, I guess, was approximately half of 2020 and most of 2021. And so now we're in a new year here, and yeah. So anybody that's watching this probably already knows who you are, but why don't you just say it anyway? Like, how are you doing? First of all, happy Black History Month. And
thank you, my friend.
How are you doing? What are you studying, etc.?
I'm doing pretty good. I'm just going through the void of life, and I'm studying chem, math, and physics all at once in USF, University of South Florida, um, where essentially they never use p-values from what I know at the moment for most of these things that they look at. Just I think it's going to be funny looking at the contrast between like the more fundamental sciences in medicine and the use of statistics.
Yeah, my former lab in biochem didn't really do multiple comparison testing and generate p-values. It was just like, you know, here's this enzyme and making like a rate curve, based on like the amount of substrate and fitting it to a like Michaelis-Menten or something. Maybe you get an R squared, and how well it fits the curve, which is probably sort of related. Maybe. I don't know. No.
No, yeah, like with regression analysis.
Yeah.
I think it would just be collinearity, which doesn't have to be significant or not.
Right. Yeah. So it's just like, yeah, how close is this physically to this idealized curve? So yeah, and that's interesting. Actually, the guy that my boss in the biochem lab, we didn't really talk about it very much because there's no point to, but he said something like I don't know if he said he doesn't like it or he just doesn't like
science that's like, null hypothesis testing, I think.
Yeah. Yeah.
I think he sort of referred to what we were doing as like discovery science,
Mhm.
um, which is kind of, I guess, like an advanced discussion point, uh, like, several points through this paper really is like, because we should start with the more basic stuff. But and so you want to eventually do research, right?
Yeah, I mean, I've already did a couple of things with like mitochondrial projects, but I do want to do more fundamental science research. I'm not as interested in the medical side of things. Maybe with genomics, but that's about it. And then, funny enough, in genomics is like there's so many tools that they have to use to get actual meaningful clinical data because it all overlaps, and it's like you barely use the p-value as indicative of anything important, just the probability that your findings aren't false positive or false negative, but not that they translate to like a clinical outcome from a gene effect.
Yeah, yeah. There's a lot of ways. Statistics do a lot of things, and
Hmm.
so yeah, I guess what I'm doing now is like, it's my lab is kind of biomedical, like regeneration, but I'm trying to do sort of, like the bioelectricity thing and more fundamental stuff, which, yeah, in my particular case, like I wouldn't really, well, maybe we'll wait to get to that. So because that's more like what kind of science, maybe there's some
Yeah.
things that just doesn't really make sense to, just intuitively to be like, hey, is this what's the likelihood of a type 1 error here?
Right. So, how's the project in general coming along? Have you seen anything cool or done anything cool with the bioelectricity?
I have not done anything cool. It takes a long time. So zebrafish labs, I didn't know this, but like with mouse research, uh, it takes a certain amount of time. Depending on whether you're breeding like some kind of genetic mutant or doing like a nutrition thing. So obviously you have to get a cohort of mice or rats that are the right age, and then start doing the thing, and then collect it. But with the fish, yeah, you have to grow up fish. They take actually a pretty long time. They take like maybe 3 months, I think, to be adults. And then mostly what's used in my lab is embryos. So you would like cross two fish, get a bunch of embryos. Usually they, looking at beta cells, they'll have like insulin, GFP, or some kind of fluorescent protein so that you can see the beta cells in fluoresce. Get the embryos, and then they also might have the NTR gene on the insulin, so it makes that toxin when you give it metronidazole. Or there's the one that we're using now that's like supposedly safer, but it does the same thing. It basically gets into the cells, and if the cells are expressing the NTR, nitroreductase, whatever, it kills them, trying to mimic that like an autoimmune type 1 diabetes type destruction of the beta cells. So you get the embryos, you wipe out their beta cells, and then under normal circumstances, say at like a 3-day old embryo might have like a few dozen beta cells, and you can count them with enough magnification. Although it's kind of hard with that many. You wipe them all out, they're all gone, and then maybe a day or two later, they've got like 5. So some cells in the pancreas, whether it's like alpha cells, delta cells, or even like some ductal cells or whatever, kind of differentiate. Maybe not, and this isn't one of the questions. Are those true beta cells, or are they just expressing insulin? Because when there's actual beta cells expressing insulin, there's like a negative feedback for other cells to not express insulin. They're like kind of all communicating together. Like enough cells are expressing insulin in this area, and then once that pressure is taken away, other cells that are maybe beta cell like. So that's like one of the questions that some people are
Hmm.
working on. And then also, which cells are they coming from? Like, try to lineage trace. Like, are these alpha cells flipping? Are these somatostatin delta cells flipping? Or just non
Right.
endocrine cells? And then, of course, the actual thing that like everybody wants is to just make to try to get it to like make all like back
Right,
to a
right.
normal amount with a drug, or maybe like some kind of knocked in gene, maybe specifically with on the promoter of a gene that would be made by like all of the beta cells, or ductal cells, or some type of lineage that's like, okay, we think these can be like induced to become beta cells or beta like cells and produce insulin if they have more of this gene running. So stuff like that. So yeah, I'm basically asking those kinds of questions, or I'm trying to set up to ask those kinds of questions with compounds specifically targeting, ion channels and pumps and stuff. Maybe some gene therapy type stuff like injecting mRNA. Um,
Hmm.
I know Levin, he does that in a number of different things. Like he's grown ectopic eyes.
Yeah, yeah.
Um,
He does it with gene therapy though. I could have sworn he only uses like those selective ion channel related drugs, and then it changes the local gradient.
for the ectopic stuff, I think he injects like maybe in the 4 or 8 cell stage of a Xenopus oocyte, um, mRNA for like those channels.
I see. Okay.
So it'll just express, it'll be like sort of integrated into the translation process
Hmm.
of that cell, and it'll survive if the mRNA also basically like the COVID vaccine. It'll
Right.
survive maybe a few division. It'll be theoretically halved every time those cells divide, but they'll be like expressing more. And then yeah, once in a while, you just get like, and you don't know, of course, where at the 8 cell stage, you don't know which one of those cells is going to be like ectoderm or mesoderm or whatever. Uh, but so you might get like an eye if you're doing something that seems to make eyes like in the tail or in the stomach or whatever.
That's very interesting to consider that at the 4 to 8 cell stage, they still don't know really if it's going to become an ecto or endoderm. But I'm assuming that the cells themselves have already all of that mapped out. It's not like they don't start when they're differentiated from 1 to 2 cells, even proliferating. So I would assume like there's still stuff researchers don't know about from that first cell to the 16 cell stage or something, where those cells are going to become ectoderms and endoderm. I wonder if that plays into the gradients too. It's like there's a specific gradient for each layer.
Yeah, definitely. That's another one of the experiments, is using the dyes to show the resting potential
Mhm.
gradient across the body plan, and that's extremely consistent across the body plan. But I don't know if there's been live imaging. I mean, that would be something I'd like to do. I don't think we have. We probably have to like borrow somebody else's equipment. And these experiments, man, it's
I know that.
like it, because it's like a whole thing that you have to like get good at. It's
Yeah,
basically like, oh, you like learn how to do like surgery or something and get good at that, and
yeah,
be able to like do it consistently or whatever like 100 times, and then you do this experiment, and then like it may or may not yield anything.
yeah. That is something I don't think a lot of people get when people do research. It's like it takes years to do it because you got to learn the methods. You got to make sure that the methods are applied correctly, and then you're forced to get good results. Otherwise, the grant goes to. So even then, I mean, that plays into like the statistics combo. It's like you're forced to have statistically significant results, even though they may not be really reflective of the entire process. It's the niche outcome of what the researcher and the team and the supervisors wanted. And right there and then it changes all of the stats that you use to indicate that the results are good or not.
Yeah, yeah, and so yeah, that gets into the positive results bias and everything. Very unfortunate situation. But yeah, so I don't even know when, like, I started doing some I'm gonna have, I guess, like at least one project that's kind of not the bioelectricity stuff.
Mm-hmm.
That's other stuff in the lab that I started trying to get these fish to express this gene Hess6, which like my boss is there's this whole alphabet soup thing that you can fall into where it's like, well, beta cells are using these genes, and if we look at like this huge data set of single cell RNA sequencing of cells that seem to be differentiating towards beta cells. Like, what are they turning up and down? And is there one specific gene that we could kind of
hone in on.
exogenously make overproduce or block or whatever that maybe would fit as a key to either knock out something that's kind of keeping more cells from going
Yeah.
down the beta cell what's it called, end point or whatever determination,
Right.
or something that could be turned up that would, induce that.
Like a master transcription factor.
Yeah, master transcription factor for those particular cells. Like, why is it that in the pancreas ends up with like this many alpha cells and this many beta cells, and like if you have a diseased pancreas that its beta cells are getting depleted, like is there, like, what can you do to just sort of tell it, like, because there doesn't really seem to be a problem. Like for some reason the beta cells are just they're so fragile. Like, I don't know if there's any real common disease like for like alpha cell deficiency.
That's a good point. I never thought of that.
They seem to be more resilient. They don't seem to be a target of I guess autoimmune or whatever the thing is that knocks out the beta cells, delta cells, and other things. It just seems, for some reason, it seems to be beta cells. Maybe it's because they do a lot of work. They're taking in a lot of things and having that firing with the calcium. They're just very metabolically active, and it makes them more susceptible to like toxins or whatever kind of just something going wrong. But yeah, it's interesting. I think even if animals don't have alpha cells, they're kind of okay, because it's like, all right, you don't have glucagon, but there's a lot of hormones that increase
Like cortisol.
blood sugar, whereas there's kind of only one that brings it down.
That's a really good point. You would think selection would have made some kind of defense mechanism for beta cells then, since they play a much larger role. But I wonder if, like what you're mentioning about calcium being a role, if that also plays into the bioelectric gradients. Like the cells that are more sensitive to ion changes will experience death easier. And then what it looks like biochemically is like a TNF alpha induces cell death, but in reality, it starts at the ion gradient in the membrane, and then that's like a downstream effect.
Yeah, I wouldn't be surprised. I mean, there's certainly, it's got to be, I mean, because that's the thing that really differentiates the beta cells, I think, metabolically from their neighbors. And I do think, though, evolutionarily, it kind of makes sense, because diabetes sucks, but you can kind of be okay. I should say, all right, if your blood glucose gets to like 20, like, nanograms per deciliter, you're like dead. But if it gets up to like 500 and stays there, like, you're going to be sick, but you're not going to be dead. So there is sort of like more of a, incentive to make sure that it doesn't go below a certain threshold, because the higher it's more of a chronic problem.
Yeah. I seen probably like a week ago, I seen some experiment where they essentially induced about 7 times the diabetic range of molar or osmolarity in blood glucose, essentially, in cell cultures. And I think it was 7 times 150, so somewhere almost close to 1,000. I think, what are the units? Millimole per microliter? I'm assuming it has to be millimole.
It's either millimolar or, I think I said nanograms. It's milligrams per deciliter.
Yeah. And essentially, even with like 7 times the higher end of a diabetic range, all it did was make some advanced glycation end products, and it wasn't even that great of an effect, which is interesting. It's like, yeah, blood sugar can be totally elevated and still not cause a dramatic difference in cell function.
Yeah. I mean, the real problem is not so much like having really high blood sugar floating around all the time is a long-term problem. But like the real problem with diabetes is that the cells, like a lot of important cells, are just not getting the amount of glucose or in the way that would keep them healthy. So it's kind of like people are looking at the sort of the effect, like the high blood glucose is more of an effect of the cells not being able to take it in and use it.
Yeah.
the way they want to. But that's, you know, the way that it's measured. So somebody asked if I tried the niacin flush yet that you did. No, I haven't tried anything, man. Like I don't know. I haven't bought, what is it? Tudka. I keep wanting to try that supplement because I haven't really tried anything new for a while. And that seems like a pretty safe thing to try for like liver health or whatever.
Yep, I like Tudka.
Not that I think there's anything wrong with my liver, but you know, I guess it could always be better. Your liver could always look better, be leaner.
We got to make that into a meme now. Oh,
Your
man.
liver could always be less fatty, look better. Why are you on a liver flush? Your liver is fine. It's very fit.
Let's see Paul Allen's liver.
Yeah.
Oh, man, those shows like an ultrasound scan.
Yeah. All right. So unless you have any other like side, you know, whatever banter.
I thought I'm assuming like most of your audience doesn't even know what you're doing with like bioelectricity and why it's important. But I figured explain it in a simple way because I have not met a lot of people that one are working on this and two really grasp the importance of it. Like from my perspective, it's essentially saying the changes in electricity around the cell are what causes changes in gene expression the most. It's not even the transcription factors. It's not even the secondary metabolites. All of them are just intermediates for full final changes in cell voltage, whether it's around the membrane or around the membranes of even mitochondria or the cell nucleus. That would suggest that, again, I mean, it makes sense. All of biochemistry is essentially based on electromagnetism. If you change the electric gradient, you can control the cell in any manner you like. So with the beta cell stuff that you're doing, you can change the gradient for alpha cells and they may trans differentiate, become from one type to another, like a beta cell. And I find that really, really interesting in the long term because it would suggest that with any genotype have a spectrum of effects that probably have never been seen before.
Yeah, it's like a different it's a different just overall theory of how the organism arranges itself, because right now the dominant paradigm for research, whether it's regeneration or anything, but let's just, you know, stick to that, is right. or differentiation is transcription factors. So looking at if an animal can you know, some amphibian that can regenerate its tail and then this other species can't, and it's like, what, when you chop off the tail of the one that can, what the cells right at that point, or maybe a little bit further up or whatever, that might be influencing it, or what's going on with the blood vessels or the nerves and what they're sending down, their factors and stuff. What genes is that animal expressing that, you know, send proteins out or just work inside cells to create a blastema to remake the tail, that the other one that can't regenerate, they don't express that, or maybe they don't have that gene or whatever. And so the current sort of overall theory is that, you know, animals lose like this gene or this transcription factor or a bunch of genes, or just kind of go down a different path to focus on this one thing, and then they lose like regeneration ability, or they lose differentiation, or, you know, crazy things like tadpoles becoming frogs, which is like a totally, like when you really look at it, how there's like some memory in the cells of the original tadpole nests, like, but you can like scramble them up and they'll still become like an okay frog, but there's some things you can do to them that will make them not be able to become like a normally shaped frog, or like the butterfly thing. The
caterpillar stage, the metamorphosis.
Yeah, it just like turns into soup, and then so this is just an alternative, I guess, alternative hypothesis. So that all of those transcription factors, see, because here's the thing, like some of the alternative science, the vibe online is they'll take that, they'll go, that's not satisfying, which it's kind of not satisfying. There's certain things that are not satisfying about that. It might take a really long time to get into, but um, the simplest one would be, how does, like what you were saying, in the early single cell to single digits of cells to low double digits of cells stages, how does one cell know which fate to move towards compared to its neighbors? And you would think like the only way to explain that with just proteins and transcription factors would be that they're kind of like diffusing out and sort of,
Right. Yeah, I see what you're saying.
so like this one, like there's four cells, and like the one that's farthest away from this other one, they're getting like less of each other's products. But
Yeah,
yeah, but it's very like hard to imagine.
that that would be subjective too, because it's in a coordinate space that is subjective to the cell. So it's like any cell can do the same thing. How does that one specific cell do it all of the time? It's
Right.
a that's we know where they are in three-dimensional space. Very interesting.
Yes. Yeah. So the bioelectricity is, the idea is that um, the cells, they have a certain set of proteins, all around the outside, and a few relationships inside with like how they manage calcium and stuff that give them certain characteristic levels of different ions, but maybe just most simply like a certain voltage, like negative something.
Mhm.
And that, and then, and when they're touching, they can sort of tell that, and they influence each other. And I mean, sometimes they even sort of share a little bit of the electricity, and in the case of like nerve projections, some other kind of cells. But yeah, there's a very even, when you get to a larger body plan, you can see a very even potential where just, and this is outside of cells firing. So there's the bioelectricity, like the way it's taught in like a medical textbook or physiology, bioelectricity is like an action potential. So a cell depolarizes, so it's negative something millivolts, maybe 50 would be like a rather negative cell. Some of them are closer to zero. They're like maybe negative 30. This changes across the body plan and what kind of cell it is. So all of a sudden, it loses all of its negative potential because positive ions rush in, and then it becomes slightly positive, and then that causes it to do usually like it's one of its primary functions, and then it goes back to like its characteristic. It kind of overshoots it and then goes back to its long running resting potential. But what we're talking about in the bioelectricity research is the difference in just the resting potentials. So the cell isn't particularly doing anything. It's just, what is its chill vibe?
That's good. That's such a good way to put it.
And that this is different across the body plan. In the mature animal, it's different, and it has characteristic differences in the developing animal, and that it seems to be a way for cells to know. So it's like all of my neighbors are like negative 47 millivolts, and a few layers down, they're like negative 50, and a few layers down the opposite way, they're like negative 45 or something. And so it's telling them like what kind of protein, what kind of genes am I going to express? And then you can also imagine that transcription factors and physical fibrous protein, structural things, and enzymes and stuff in the cells, they all affect the potential. It's not just channels and stuff, because the proteins, like one of the basic functions in the cell is to just hold on to potassium ions,
Right.
adsorb to the protein. So if you have different proteins, so if there's a whole, a tissue that has characteristic expression patterns, it will just tend to have similar resting potentials. And so it's like this, whether it's transcription factors telling it to make this protein, or telling it to some long chain of like, make this transcription factor, which tells it to make this transcription factor, which sometimes when people talk about that, it's just like, but
Right. Yeah.
but in the end, you're producing proteins and they're having an electrical effect. So it's like, and I would imagine there's similar to transcription factors and things that like make the DNA more likely to be read and translated in the cell. There's probably electrical stuff that maybe
Yeah, definitely,
does that.
definitely.
I haven't really seen anything specific to that. But anyway, so the point is, and Levin says that the proteins, all the structural proteins and the things that would be transcription factors and stuff is sort of like hardware, like computer hardware. So like you have RAM and a processor, you know, GPU, whatever. And then that runs software. And so the electrical potential would be like software that's running along the hardware. And so it's not a perfect analogy, but I guess the idea is that right now the dominant paradigm is to try to like jam in a piece of hardware, like
Hmm. Gene
yeah, with
therapy.
gene therapy or with a drug or something to like kind of just make this cell like, I want this computer to do this different output, so I'm going to like put in a second processor or something. Um, and you know, whatever, maybe that works on a certain level, but perhaps it's just like totally the wrong direction.
Exactly.
And
Right.
that if you
Right.
change, you know, on a different dimension, the software that it's running, it'll kind of reorganize itself, even change its own
Right.
hardware or like how it's running it. Uh, so yeah, so but the problem is, I mean, for me, like Zebrafish, it's not the best model system. I mean, the frog is nice, I think a little bit nicer, um, but not that many people are working with it, so that's good too because it'll be like easier to publish if I do get anything. So yeah, I have a chemical library that I ordered from some like Italian company. It's got like 80 drugs or something that target different ion channels and things. So I'm going to do some screening for like ectopic beta cell production using those drugs, and maybe also for the regeneration experiment, like the beta cell ablation and then
Yeah.
regeneration. Um, and
And your hope.
I think Levin said he'll send me some RNAs for like channels and stuff that but like he, you know, for him, it's a little bit easier because if he just injects something and the frogs make some wacky organ in the wrong place or whatever, it's like that's great. But for me, it's like, you know, how am I gonna get like I want a second pancreas, you know, like if I want ectopic, like
I
I've
get
got
it.
like a small target.
Yeah,
Um,
I gotcha. Which would make it difficult because you got to find out the rough tissue voltage across the pancreas and try to mimic that with a drug. That's difficult. Yeah.
yeah, and like during development, I'd also like to do some dye experiments, but those are, I mean, these experiments are no joke. Like to the dye you can do qualitatively. It'll, you know, just with like a fluorescing dye that enters the cell more or less depending on its voltage, so it'll just give you a readout that way visually. But then to get quantitative results, you have to do that, and then at the same time grab individual cells and do electrophysiological
Hmm.
clamping
Hmm.
to calibrate. So like in the fluorescent images that you have, this much, you know, say it's blue fluorescence or something, this much blue means, you know, -70 and this much means -50 and stuff like that. Which I mean, that's a whole like learning how to, I like there was an electrophysiology rig in my PhD lab, but I never used it. I just watched, there was a postdoc that was like his thing. I think Ray actually did some electrophysiology.
Yeah, I remember reading a lot of his stuff on that.
Yeah.
That's what he talks about with like progesterone being regenerative and estrogen changing tissue structure. It makes a lot of sense because I mean those are the things that Levin uses for his stuff.
Yeah. So, I mean, maybe this will be like a pretty long
I
haul
can, yeah.
project. Or just, I admit, just, you know, I mean, like when you're in science, and this is one of the problems, like you have to publish something. So like you know, I'll have other stuff going on too. And, you know, it might not end up, you know, working out or whatever, but I truly believe in the alternative hypothesis. Like I believe that if you could get, you know, a cell that has some type of progenitor, beta cell progenitor
Mhm.
affinity, if you could target that bunch of cells or whatever, their resting membrane potential, that would be the most powerful inducement more than any one or several transcription factors that perhaps are not so related to the bioelectricity. That the transcription factors are probably lagging indicators. They'll start getting made after you tell the cell, like, you know, here's where you are and here's where you're supposed to go. And then they'll be like, okay, well, I got to make this, and this protein to get there. So.
Yeah, that if that were done mass scale, you would essentially reverse all type 1 diabetic issues.
Yeah, I mean, if there was like a drug, the other thing is, of course, I mean, if you're taking a drug, it's in the whole body. So like, and
yeah, that's a good point.
It's everywhere. Well, channels are like no joke. So like, there could be a certain thing that you modify in certain cells in the pancreas that'll have that outcome. But like, I mean, it's really easy to kill cells or make them diseased by knocking out channels and stuff. So that's where like the gene therapy, but obviously you can't really, like, the gene therapy is nice when you have like an egg, you know, and you're like, I want to just give this whole thing a different expression pattern. But once you have like a developed animal, it's a whole other issue. Like the BioShock plasmid product. I don't know if you ever played that game.
No.
Yeah, totally predicted the COVID vaccine. BioShock. I think basically the guy that wrote the story, he was just taking like the CRISPR stuff as it was being talked about in like maybe 2007 is when the first one came out, and just made that a gameplay mechanic. And in the lore of the game, there's these like snails or something, like deep sea snails that express a protein that can insert genes, and so people make these products, and it's a way you get like abilities in the game, like you insert, you know, I can shoot electricity out of my hands plasmid.
Right. That's very interesting. I'm gonna have to play this game. First of all, I gotta get a console because I don't have anything here.
Yeah. Oh, samesies, man. I mean, you can see like what's behind me right now is basically my living space. All right, so thank you for inviting me to talk about my research.
Right, I want to make sure people understand, like, you're doing some cool stuff, and I'm sure when they hear us talk about like pancreas and beta cell transcription, et cetera, bioelectricity, they don't really know what's going on. So I figured it'd be good for them to understand.
Yeah, and yeah, if I have any interesting results, I mean, I'm definitely gonna talk about them and whatnot. But yeah, I actually, I'm gonna. It's like it's a tough thing, because when I have a project that could potentially be interesting like that, your job starts to. Because what's nice, like if you want to make internet content or like write stuff about nutrition, what's nice for that is to have a boring job that you just do for a certain number of hours, and then after that you get to be creative, like outside of. But everybody's like, oh, I want to be creative at work, and that's cool, but then like it's hard to be creative and interested in stuff like if you're already doing something
new.
Yeah. So, but I mean, it's hard to complain about having like an interesting potential project to work on.
Of course. The job of a scientist, a researcher, is so interesting. It's like that is the purpose. We want to discover new things.
Yeah, and like some of the things I've done before, the project is kind of set out, and there's a certain amount of creative input, but at a certain level, and this is true with anything, but a lot of projects are more like you're kind of walking into just a bunch of tasks you're going to perform.
Yeah, I gotcha.
And then there's the other end of the spectrum where like you have to make a lot of decisions and think of a lot of creative solutions to like technical problems and stuff, or like crazy ideas that you can pitch to somebody that don't sound crazy. Like just walking up and being like, hey, let's do bioelectricity. Like that doesn't fly. You have to kind of have like, look at this paper, and you have to use different language, that would make sense to a person that wants to hear terms like transcription factors. They don't want to hear like certain things. So you have to, but I mean, people are open to this stuff. You just have to kind of use their language. Like you can't make it sound like woo woo, or else they'll they'll get like turned off by it.
It's like the art of grant writing. I don't know if you're doing like grant writes, but I from professors that I've had, they talk to me about it's like an entire 6 months of just learning how to talk the talk so that you can convince people to give you money to do the stuff you want to do.
Yeah, I mean I don't I did I've done one, like I wrote a section of one grant when I was in graduate school for my boss at the time. I've written like practice ones like grant writing courses, but I've only written one real section, just one section, and it really sucks. Like just outside of because it's not a human interaction. Like if you're working under a scientist and you want him to like buy something so that you can do an experiment and you have to sell it in a way where he's like, okay, this is worth the time and money and stuff. That's a very human interaction. Grant writing is not a very human interaction. It's like you're writing to a committee. They're it's super conservative. So like and it almost makes it. I think this is actually one of the main criticisms in the economics, the Terence Keeley, the economics of scientific research. He says that sometimes science is so conservative, like they want you to present potential experiments that have such a high chance of success that they can almost not be called experiments. It's just like
Yes.
the next step in a like, oh, we gave this drug to a rabbit, let's give it to a dog, and like it'll probably have the exact same effect. And they're like, that's a great experiment.
Right. Yeah. That plays into that Nature paper, or it was actually like a blog article in Nature. They were saying that we have all this new data, but we don't have anything novel, or anything that has made a breakthrough in many scientific fields. And it's, I think, because of the way that academia is run, it's like you don't really get the icebreakers often, like Michael Levin stuff. I don't know if it's because he's at Tufts University in specific that he gets a different kind of support, but I don't think that would fly in most universities like Stanford or Caltech or some other high level profile of university. So it's like you have all this data and they're very similar to one another, but nothing important comes from the data.
Right. Yeah. It becomes kind of like, yeah, there's a lot of things that
Mhm.
could be said about that, but yeah, yeah, it becomes like it's kind of like certain types of like modern artists that make art for other modern artists, and
Mhm.
eventually it's like people are like, what am I like? This isn't, and it's like that. It's like certain researchers. It's like, oh, we've got this system to model human disease, and then it becomes about studying the system, and then it becomes about studying like a little piece, and it's like this is like losing, like who really cares if this model organism, you're like off studying things that are not analogous to the human system you're trying to model anymore. You're just kind of like trying to ask vague questions, like, yeah, just yeah. That's one of the big problems, and it's almost impossible to stop because a lot of those like really technical things where it's like, okay, we're going to totally figure out this model organism, like people in the field, they kind of like that and they respect that, and like with regeneration, it's like, oh, we want to like help people who get their hand cut off or something, maybe be able to regrow a hand. Like, wouldn't that be awesome? And then eventually they're just like super specialized in how exactly this subspecies of toad like regenerates their arm, and to the point where like it's totally like losing the plot from anything that they're just getting farther and farther away from the human
Right.
outcome eventually.
Right.
All right. So, and part of that, p-value has been part of that transition and is resented for it. So the paper that we're having like a journal club here. It's a little journal club. And we are reading a little bit. We won't read the whole, we're not just going to read it, but discussing a 2017, looks like, paper by just two authors, which is always nice. Well, it's not a research article, so sometimes, you've got these like 20 author research articles. Denise, these are, okay.
He's like a
These names are Well, okay, John P.A. Ioannidis.
Ioannidis,
Oh, Lowen.
he's a statistician from Stanford, and he's the guy that essentially wrote the replication crisis paper that came out in like 2005.
Yeah, yeah, he is hardcore self citing in this paper. So just from
That's
that,
true. That's true.
just from that, I was like, okay, so um, this guy is like single handedly, he's like on a mission here, and he's writing these papers and then writing more papers in which he's like seems to be the one of the primary critics here of the whole null hypothesis significance testing. So this is when null hypothesis significance testing is unsuitable for research, a reassessment. So
Should we explain to them like what that even is, like why it's used in research? Because I'm sure they think there's only one type of statistics, and that's you prove your hypothesis wrong. And it's like, no, that's not the only way you do stats.
right. So and he does go through the history of this, which is very interesting. But basically, the what is maybe sort of considered, not by everybody, but the scientific method right now is where you have some type of manipulation or experiment to get some type of result, and you sort of call the idea that your proposed phenomenal causative agent in this result that you suspect to get, um, having that kind of a cause or having that relationship is the alternative hypothesis, and then the null hypothesis is sort of set to that's not what's going on. Like whether it's hey, we think that such and such causes cancer, something in the water, so go collect a bunch of survey data of people that are getting maybe a certain type of cancer specifically, and comparing that to, if they live in a place where this thing is in the water, and then people that get that cancer that don't live in a place where that's in the water, or people that live where that's in the water and don't get cancer, and to take all those data together and see whether they're statistically significantly different, such that you can say there's likely to be that thing in the water is likely causative of that cancer. But technically, very technically, what you're testing is the likelihood that the differences you're seeing in any of these populations that you're looking at, sampling from, could be due to just chance. Yeah. So if you take like 1,000 people from one country and say that country has on average taller people, and you take 1,000 people from another country that for a stone cold fact has shorter people, like you've surveyed everybody in the country, you could get 1,000 people, a very large sample, a respectable sample that would have good power and all of that, where they aren't different, even though they're different. Or you could even possibly get the incorrect relationship, which would be a little bit more extreme. But and then so that would be a false negative.
Mm-hmm. Type 2 error.
Yeah. And then the flip side would be, let's say there's two countries and actually the average heights are exactly the same, but there's a certain chance, depending on what sample size you have and the variance within the countries and everything, that you'll get a big old sample that should be enough to have an idea that will say that the heights are different, and that would be a false positive.
Mm-hmm.
Just like, in cancer, a false positive would be like a cell that you think is cancerous. It's a little different because that's not so much statistical. That's more of like a methodological.
Yeah, right.
But yeah, so because in cancer testing, you can't really have a false negative because you can't just test every cell.
It becomes a continuous variable, so it's not like a discrete outcome.
Yeah, like you can't have evidence of no cancer. You can just have no evidence of cancer.
Right. And that's where the hypothesis testing comes in. When you get a result that suggests a true positive or true negative, meaning that that's the correct outcome, it's not saying anything about the outcome itself. It's saying that the experiment performed therein has a correct outcome, but you can't just take that information and then translate it to everything else as a scientific fact. And with null hypothesis testing in specific, to do so, you would need to do it a bunch of different times, thus replication with scientific methods. And that is the frequentist approach. You do it a bunch of times, the frequency of the test is performed over and over. And even then, there is a limit to how accurate you can get with higher frequency. Like with large data sets, you lose the significance clinically, for example, even if results are really statistically significant. You're bound to have a lot more of these p-values just by distribution alone become statistically significant, but it doesn't mean that the outcome, again, is important. It can be totally unimportant, or it could be totally unrepresentative of the population that this is studied. So in a like really simple way, p-values themselves tell you nothing about the scientific fact or the hypothesis you're investigating. It just tells you whether you can reject it or not. And rejection doesn't mean that it's false or true. It just means you rejected it.
Right. And originally, and so getting into the history of where these things came from, at least according to this author, we get the p-value from Fisher in 1925. So, and he basically came up with the canonical used p-value of less than or equal to 0.05 with
arbitrarily.
Yeah, and there, as far as I know, there is no way to mathematically justify it. There's no rational reason to say. So a p-value of 0.05 means there's a 5% chance that you could get the same results or more extreme from chance.
Yeah, it would, because the equation to determine p-value depends on the sample chosen, the mean of the sample chosen, and then the variance or the standard deviation. It's like 0.05 could be accurate for any number of conditions as much as it can be accurate for inaccurate for any other number of states. It's like, it's really hard to say what is categorically true of a statistically significant result. It really depends on the subject chosen and so on. Like in physics, they use 6 sigma values. So you have to get 0.000005% or 1% probability as a p-value to actually determine that your experiment is correct. And by correct, I mean close enough to correct. It's really dependent on the fields.
Yeah, and if I understand, 6 sigma is roughly equivalent to like something would have to randomly be rolled.
I think 1 billion times over, something like that.
It's something like the life of the universe or some.
Yeah,
I remember like hearing some comparison, like how many atoms there are in the universe, or maybe not that many, because that would be, but it's some kind of enormous. It's like the least likely thing.
Right.
Or maybe it was something that would by chance happen once. Yeah, and I think in the supposed lifetime of the universe, so however many billion years, like a 6 sigma event would be something that you could expect to possibly happen randomly over that amount of time. So something that can't, and this was made famous when I think one of the financial collapses was the savings and loan. One of the not the 2008 one, but one of the ones before that. There was I think Long-Term Capital, some company that hired a bunch of like Russian mathematicians and all to do all this sexy math and come up with how their investments like can't possibly fail and and then all their investments failed, and they had all calculated like whatever happening would to be a 6 sigma event. So obviously it wasn't a 6 sigma event.
Yeah, the models that you use can inaccurately present something that would instead be expected, even though your models suggest it's unexpected. It's like the models play a role in getting the results statistically significant or not, which plays into what this is. It's like hypothesis testing. You're likely to get p-values that are really low, and so it would suggest that the null hypothesis is rejected. But really, when you do other methods, you find completely different results, and there's inconsistencies with the methods, and so likely the results of the studies are not translatable. So I'm assuming like with these Russian mathematicians. They used a good model that can support the outcome they wanted, but it wasn't representative of the true outcome in general, reality.
Yeah, yeah. It's I forget exactly what they were sort of saying was so unlikely. Maybe it was something like the chances of, say, you've got a basket of investments, like 8 of these companies going out of business is a 6 sigma, grind set.
Yeah.
And but maybe they didn't take into account, and this would be a really simple error, and this might be too simple and too stupid for them to have made, but one error you could make
Right.
would be to make them independent variables instead of a model where they're dependent. So like maybe those 8 companies, maybe a couple of them have relationships, so if one of them fails, it would radically change the chances of the next one
Right.
failing. And then maybe the first one didn't have a relationship with the 5th one, but the second one did. So the first one and the second one have a relationship, then the second one goes down, now the 5th one goes down. And so what could have been a 6 sigma event is like a likely
Inevitable.
event. Yeah.
Yeah. Yeah, that's literally Bayes' theorem. It's like you're treating them as independent variables when in reality they matter. And if you know the relationship exists, you can get a much tighter likelihood of getting a true result that you would expect. But with frequentist approaches and null hypothesis testing, you're almost forgetting that, or you're saying it's totally unrelated, and we're getting a perfect sample from the perfect population to get the perfect outcome. And I don't think that's accurate, especially in medical research.
Yeah. So Fisher's idea, and I guess that maybe this was an improvement. I mean, I've read some old science, and what's usually nice about it is they don't have a lot of statistics. I mean, it's kind of like,
Yep, that's interesting, huh?
yeah. I mean, I think about it like you don't really need a sophisticated statistics to, say like cyanide is poisonous, you know, um, and then sort of figure out like what binds to what P450 or something. It just doesn't sign.
Uh, Cyanide goes to mitochondria, cytochromes. Man,
Yeah,
I forgot.
yeah.
Something.
It works up respiration. Just like a much more powerful, I think, carbon monoxide. It's like the worst.
It binds to iron at cytochrome C
Okay.
oxidase, and so it stops the action.
Yeah. So and I guess maybe what you would need statistics for would be like what dose what's the LD50? What's the dose? You got a bunch of like, you want to use animals or whatever. They have a certain weight. How many grams per kilogram weight? Um, can you give these representative animals such that 50% of them die? And then you could kind of say
Mhm.
and then that might require you sort of getting into like, because then there's all kinds of variability. Because obviously, if you just like and this but the funny thing is we know cyanide is poisonous, so like we know that as a fundamental truth, so we already know that there is a dose that will kill basically any animal, right? It's just a question of what the dose has to be.
Mhm.
So then you can start to ask the question of how much, and that could give you insights into what the mechanism is or whatever. But in research, you don't know often whether the compound that you're looking for a certain effect causes that effect. Like we know certain things are poisonous. It's just a question of dose. Like if you give, the LD50 to an animal, might not die, almost certainly will be the worse for wear. But if you just give it that dose again or 3 times, it's going to be dead.
Yeah.
No problem. But with a drug that's like, okay, we think this drug might lower blood glucose in diabetics. It'll be like an insulin potentiating factor or whatever. You could start asking the question of dose, but you don't even really know whether it works. And then of course with drugs, there's side effects is kind of a different thing. But in research, in like kind of like real research, you're sort of asking questions where the true factor, it's kind of like what I was saying about countries. if you don't already know the average height in a country and you're taking a sample, that's a much harder, because when researchers set, their alpha, acceptable alpha, or they take the alpha of 0.05 and then set an acceptable sample size based on the power that they want to achieve, the beta, and so forth, those are all based on, in some cases, nothing, because if you don't know what the true value is, because if you take like Stats 101, you'll calculate those things based on knowing. So like if you know a population has this true characteristic and the population is this many people, how many people, what size sample would you have to take to get the correct result from that sample, with a alpha of less than 0.05?
Yeah.
So, but that's not research, because that's asking a question. That's asking what kind of statistics, what kind of numbers need to be plugged in such that like running this simulation gives you the correct answer, and we know the correct answer.
Yeah, exactly.
And that gets into the second bit of history, the Neyman and Pearson. I don't hear Neyman's name very much. I hear like Pearson coefficient. I hear his name a lot. Maybe it's one of those situations like sometimes you see two scientists and then like the years, start coming and they don't stop coming, and then like the one guy's name just kind of fades away.
Yeah, Neyman, I've come across, but no, I can't put like a thing to it. You always see the stats book, it says Pearson on there, or like math courses, it says Pearson for your course material and stuff. Never Neyman. That's true. That's a good point.
Yeah. So they came up with certain parameters for type 1 and type 2 errors, and they're good parameters, but they were actually working in a system that, as I was just explaining, they knew the outcome they wanted. So they were not, they actually, so Fisher developed his p-value procedure and the kind of underlying assumptions and his, a 1 in 20 chance of a random, that's good enough, whatever, like we'll just kind of go with that. He did intend that for research, but he intended it for, as you said, replicated things. So something that you would do like many iterations of, maybe different people would do. And then if it continues to hit below that 0.05, we could feel good about that.
Right. And he didn't say like it's a established scientific fact. He just said there's a good confidence here that, hey, look, this is what you'd expect, essentially. It's not like this is the truth and nothing else exists outside of this.
Yeah. I guess one way you could say is we can move forward in this field with
confidence,
tentatively, but continuing to maybe, check back and ask this question in different ways.
Mm-hmm.
And then this also gets to the asymmetry. And this is what's nice about Fisher's idea. There is an asymmetry to it, which is you never actually accept. You're supposed to not really accept the alternative hypothesis. You're supposed to not reject the null hypothesis, which is, because that's another thing about science. Science likes to, things like theories and these words, they like to present it as this isn't what we believe. This is just the least unlikely thing according to the state of the data, like that kind of. Somebody just said they can't hear me.
Right. Let's see.
If that's true, I'm gonna lose it.
let me play it. Hold on. Somebody just said I could hear you just fine. Let's see.
I could turn up.
That's true. I'm gonna lose it.
You hear yourself, right?
All right, I'm gonna turn up a little bit. So right now my gain on my little interface here, I go out of my way. I had to buy a second interface, but an XLR to USB thing, because I left my other one in America, and I have a decent microphone. I just hope everybody knows that I'm trying. So okay, so Neyman and Pearson, I forget what industrial application they were actually using, but the author gives an example of industrial quality control. So they developed their methods for not for research, but for industrial quality control. And the example given here is if a factory is producing screws, screw heads, there's a certain acceptable size, you know, that you think is acceptable, or like you have a basket of them, and only, you know, what number out of, you know, 1,000 in this basket, is it okay that they're this many millimeters or whatever off from the exact thing that we put on the box that says how big they are. And that's something where you just kind of decide for yourself. Like, you can get real world feedback from customers, you know? So there's like, if customers start complaining, then you probably have to tighten up your error rate. So you have to like, decrease your alpha essentially. And then of course, as that's happening, there's another rational metric, which is cost. So maybe to make them this many millimeters tighter and this much less likely to produce an aberrant screw head costs 20% more for the production process or something, or doubles the cost of the production process. And you can imagine, because somebody might say, well, why not just make them perfect? Why not just make all of them perfect to the point where you're measuring, whatever you're using to measure them can't even distinguish the difference? Well, maybe that would make them cost 100 times more, because with a lot of...
You're covering for a systematic error there. It's like you that costs something. You can't just make the measurements tighter without losing value otherwise.
Yeah, and with physical processes, like there's a point of diminishing returns. Like you might, with these kinds of things being made in a factory, you could make a curve that's like, okay, so we could make these things and they'll cost like a 1/100th of a cent each unit, and here's how many of them are not good that we have to throw out. And if we double, change the process, input materials or whatever production process, maybe it costs twice as much. So that's still pretty cheap. And now it like cuts down by half the amount of bad ones that we get. That's good. But then there's a certain point where it's like, we increase the production costs by 20 times and it only reduces by 1%. Now you're getting into the zone where like, if you show that to somebody, they're like, well, that's not worth it, right? So you'll notice that all of those things are things that are knowable. So I guess in the factory, before you're actually making the screw heads or whatever you're making, you don't know what's going to come out of the machines, but then you do it. And it's like an experiment that's being run permanently and continually. And you're getting millions of units maybe, and you can take large samples and look at them. And you can set rational calculations, like we were just saying, for cost benefit analyses. It's actually like the in Fight Club, the formula that Ed Norton character uses for the car crashes. You remember that?
Yeah, I've watched it literally this summer. I just don't remember what the exact formula is.
It's like the chance of a catastrophic auto failure such that it'll cause a fatality times the costs of an average out-of-court settlement. And if that number is lower than the cost of a recall, they don't do one.
Right.
So basically what he's describing there is, setting your, you could say, all right, how would we analogize that to like alpha and beta? And
so the alpha is the car crashes that you'd expect. If you have a couple of car crashes, then your null-hypothesis is your car is safe most of the time. But even then, you wouldn't accept your null-hypothesis. It's just a statement.
Actually, it's not that it's safe. It's that it will not cost. It will
right, because you're still
not be cost efficient to recall.
And then the beta would have to be what the power, the sample of cars that you test this with. So you're looking for 90% power, so 10% fault or type 2 error rate. Then that would be your beta. It's like you have to have enough cars to make sure that this is true 90% of the time.
Right. So yeah. So basically, because in that case, the rational outcome is saving money. in the case of what Edward Norton is claiming, you know, his character is claiming is the point of his job for this auto manufacturer, a major one. So they don't really care if like one person, their car seizes up and incinerates them.
Right.
What they care is, okay, we have to give that family $2 million, but it'll cost us $20 million to recall all the cars that have like the chip module in the such and such that failed, that caused that accident. So we're not doing it, kind of a thing.
Yeah.
And so all of that is to say that none of those things are really knowable in research science. So you don't know, you don't already have an outcome you're shooting for. You're like throwing these numbers that are supposed to be sort of empirically tested against like a rational calculation, something really hard like money or just amount of bad screws or whatever, that I don't accept more than 1% of bad screws. I will not accept that. So we have to, you know, change the process if we're getting that much. Yeah. So in science, you're just like looking into the void with these numbers and these methods that are designed to look at something that's like not in the void. And that's basically the history of these numbers. So what then happened over the course of the 20th century is p-value and all the other factors of null hypothesis significance testing took over, most of biomedical research. This paper is specifically, I don't know if this guy, you probably know, is he a neuroscientist? Because he keeps referencing neuroscience.
He's a medical doctor. He's an epidemiologist mostly, but I'm not. I wouldn't doubt that he's gotten into neuroscience because, yes, I seen he was talking about fMRI data.
Yeah, he was giving that as like, that's the worst. So he was kind of saying the fields that have been most penetrated by this and that are the most poorly served by null hypothesis significance testing are first and foremost neuroscience. I've never really done neuroscience, but fMRI activity. Actually, I did just watch a video where this girl was talking about people were getting significant MRI results from like dead fish showing that they're alive based on MRI scans because there's a certain amount of noise. So like you could just have a bunch of dead bodies, and even dead bodies are going to have some type, whether it's just machine noise in the fMRI or they have some, for a while after they're dead activity, which does not indicate life. And then conversely, the ones that are actually alive might have lower than average activity. So there's like all kinds of noise, right? And that if you just kind of blindly throw these numbers and do this research, like you're just like a graduate assistant or something, or an undergraduate or whatever, and you're not thinking about it, you're just like scoring, you're just taught how to score a thing, right? Like an image or some kind of output, you score it, and then you tabulate all your data, you throw it into the analysis of variance, or you do like a t-test or whatever simple thing, and you're like, hey, look, these two are significantly different, and it's just a bunch of dead fish. How embarrassing.
Right. And the problem there is even translating it as a outcomes, like statistically significant. What the experiment is statistically significant. It doesn't say that the fish are alive. It just says, hey, your MRI machine has this data, and once again, completely
exactly
angled by saying, okay, then naturally the clinical effect of this is logically the fish must be alive. It's like, no, totally different things here. That's just your data that the p-values are specifying. You can't extrapolate the data and say, okay, clinical effect says this is significant, so it's here. It's like, no way, that doesn't work.
Right. And actually, so that gets to, because we were saying before we went on air, the paper, it's very interesting. I actually might not recommend that this be read by the audience, at least not the whole thing. I had trouble reading the whole thing. I kept getting bored because I felt like a lot of the points were the same points over and over again. But it has these headings like p-values are random variables. So that's kind of what you were just saying. Like a p-value is just a mathematical representation of.
it plays into what you were saying before as well about knowing things. Like statisticians know where this sample information that they're choosing comes from, and that plays into probability theory. It's like for probability theory to be accurate, you have to have a set of an outline of the sample, a sample space is what they call it, and an outcome you're expecting from that sample. So there's a one-to-one correspondence. But in medical research, there can be multiple correspondences between different things. Like, let's say that one drug, metronidazole, has this one effect. Okay, cool. We see that effect. It's statistically significant. So the drug is working. It's like not necessarily the effect without the drug working because it's working in another manner. So it's not a one-to-one correspondence. It's multiple different pathways influenced by the same drug. And I think that's where they mangled this stuff up. It's not representative of where this comes from, probability theory. It's just taken as a surface level information that you can translate clinically when it can't be most of the time. For that, you need, again, Bayesian statistics to get close to a causal relation. Like you can't denote causality with significance testing. It's not right to do so.
Right. So I have the Bayesian alternative hypothesis computation as one of my notes for like things that people are trying to use to solve this problem. So we could probably maybe go over that. So the p-value, just to super clear, it only computes how unlikely the null hypothesis is. So it doesn't compute how likely the alternative hypothesis is, obviously.
And it doesn't also state that the null hypothesis is wrong. It just says the observed data is unlikely. It can be wrong or right still, but it's just like the observation itself.
Yes. Yeah. Yes. Actually, the current observation of the null hypothesis. What it basically means is you have a distribution. So P can be 0 to 1, and if you have something that is a true null not effect, you would expect, if you did like 1 million experiments, for the P that you get to just be randomly distributed across 0 and 1 over time.
Yeah.
And I think they mentioned that as like P curves as a way of trying to leverage like bigger data, master P, if you will. And whereas if the null hypothesis is true for real, then over the course of many experiments, you would expect the p-value to be skewed towards 0, just skewed. You can't really say it's going to be any specific number. It's not going to be 0. But it's going to be like skewed towards 0, not symmetrically distributed across 0 and 1. So you can see how just saying if it's below 0.05, then we say that it's true. The alternative hypothesis is true. So you can see how arbitrary that is, because it's kind of like saying, electrons have this cloud of like where they may be, and then being like, hey, we spotted an electron in this one place, so like it's definitely in this orbital, because like trying to like pin something down that's more of like a probabilistic cloud of things going on. So the Bayesian, I actually don't know how. So normally with this stuff, like you enter it into a computer and it spits out, whether you want a p-value or you want a Bayesian test or you want some kind of p-value with a post hoc analysis, to tighten things up or whatever. We've long since left the realm where like human beings are going to sit down and like Goodwill Hunting, like do that equation themselves. So I actually don't know how the Bayesian math works, but it purports to give you information about both the, likelihood that the null hypothesis to be rejected, and also the alternative hypothesis to be accepted.
The opposite event, essentially. And each time you take, so with Bayesian methods, each time you find a result, heads or tail toss, you got heads in 40% of the time, tails 60% of the time. Okay, that is one total event that you have information about. Then you repeat the experiment, and now it's 50% of the time, 50% of the time. That's another event that you have information about. And you do this over multiple periods of times, you have a substantial amount of information that you acquire from prior experiments to state with a very strong likelihood ratio, that's what they use specifically, that the outcome is, okay, 40% of the time it was heads, then it was 50%, then it was 45, then it was 55, 50, 50, 50, 50. It's like, okay, we can imagine based off of all of the a priori information that we got, it's not a rigged coin. There's 50% heads, 50% tails, roughly. That's how Bayesian statistics work. So it's like with medical outcomes, you know that the person, for example, that has a heart attack, the person is having signs of low O2, their oxygen saturation is down. Okay, likelihood goes up that this person has a cardiac issue. You then see that their echocardiogram shows ST elevations or something's wrong with the ST segment. Okay, it suggests specifically coronary issues. Then you do another test like what else? A heart scan, or actually contrast agent in the coronary arteries to see if it's dilated or if it's contracted. And then you do see that it's actually contracted. And now you have a 99% likelihood ratio that, hey, this person is having an acute coronary event. That's how Bayesian statistics works. So with medical practitioners, they don't even realize that what they're trying to do is take Frequentist approach with null-hypothesis testing and translate it to Bayesian approaches with prior knowledge building up into a very strong confidence interval of what you think is happening. But those two are mutually exclusive. They're not one and the same. And using a p-value to try to get that likelihood ratio does not translate equally. And that's where they're coming up short. So with medical research that's statistically significant, they're not even realizing that they're trying to apply different methods to one about.
Right. So like it would be silly to just, do a big experiment of like how many people have this thing happening while they're having a heart attack and how many people have this thing happening and they're not having a heart attack. Like and get like a ratio and a p-value for it, and then somebody's having this thing happening and be like, are they having a heart attack or not? Yeah,
exactly. It becomes silly too, because, for example, let's say you have a population that you're looking for heart attacks, and so your hypothesis is based off of you want to see a heart attack. Well, then you would expect the p-values to be non-statistically significant because everybody would be evenly distributed. So everybody gets a heart attack. The p-values would be at what, 50% roughly. So in that case, the clinical outcome is still important. I mean, the person is dying, but would you say that it doesn't matter because it's statistically insignificant? It's like it's silly.
Right. Yeah. It's that they're they're dead, but their values of whatever O2 saturation were not statistically significantly different. So
right.
Right, exactly. So, I would look at that as like, you kind of want to get outside of what you're looking at and kind of, look at it from another angle instead of just like collecting more and more and more big data. So that's the one thing that people can do. And it seems like from my perspective on the inside, that mainly the reason why everybody doesn't just have to do that is because it's like an inertia thing. Like everybody just publishes, they have a publishing record using this statistical analysis, and they, for RNA sequencing data for, up and down regulated genes, they hit it with a certain statistics to try to get out false positives. And that's it, and they just kind of go from there, and then they'll just take and look, this gene's upregulated statistically significantly or whatever from the treatment group. And the other suggestion that I saw floating around, which I think this sort of gets into some of the problems of more complex experiments, but two-step publication. So the idea that you go to a journal, that instead of journals just being recipients of a final manuscript, you would say, maybe write up like basically a research proposal and say, I'm going to do this experiment. These are the expected outcomes. I think this drug can do this or whatever. And then they would agree to publish your paper before the experiment is done. So there's no, we tried to do the experiment a bunch of times and then it finally worked kind of a thing. It would just
Right.
be like, this is what
Yeah,
we're going to
final
do. Yeah, one time, and we're laying out the parameters beforehand. We're not going to do it and then go, we
Right.
need more power, so let's do it again with a higher number. Like you
Yeah.
lay everything out in the first place, and then somebody actually from the journal would assess the science, like the idea, instead of just the results that you got. So like the idea, like you could have a crappy experiment and just get lucky and get statistically significant results, and then that would be publishable. But
Exactly. That's publication bias.
right, so that's the and okay, so that to me seems like it, but I don't know if that would work for, like, I was giving the example of like a surgical. Well, no, it would work actually, but you would have to, again, you would still have to do multiple experiments. Like you would have different groups, and they'd say, oh, we're going to repeat this and we're going to do it this way. And we're going to do, and it would just be more open. And I think a lot of people wouldn't be so into that, because they do. And I understand it. Like the experiments that I'm trying to do, like I'm pretty sure that some combination of ion translocation modifying drugs or gene therapy or whatever can cause de-differentiation and redifferentiation of cell type A into cell type B, at a certain rate or whatever, a certain likelihood. But, if I do one experiment, I use 20 micromolar, and then I use 2, and then I use 200, and then it's like, do I have to count all, like if if it's a failure, it's like there is a certain point where like you can have an experiment fail and the phenomenon be true.
Right, exactly. That is collinearity. Like I was mentioning with like sample space and probability theory, it's never just a one-to-one correspondence with things. You can have multiple things influence the same outcome. So when you do tests for things like that, you would use like principal component analysis, multivariate regression models, and then it's more regression analysis than it is p-values, from my understanding, if I'm not mistaken. So it's like it doesn't even matter what the p-values are. It's like you're just looking for relations between things, and if you get the relation you want, you know that your results are important. But it doesn't mean anything if it's not statistically significant or not.
Right. So, yeah, I think like different fields. That's the other thing. Different fields, like when I was doing biochemistry, there's very little statistics, and when you're doing molecular biology, there's a ton of big statistics.
Yeah.
And a lot of, frankly, like p hacking, which we didn't really mention, but that's kind of like the derogatory term for, imagine you have some data and you feel very strongly that there is a maybe modest effect, but you think there's a real effect of what you looked at, but this, your p-value is not achieving an alpha of 0.05 with the statistical test that you choose or the post hoc analysis you choose. Because there's, like Bonferroni, there's a few different, that you can just plug into if you have like R or, I usually use Prism, GraphPad Prism I used to use, and it'll just do these tests for you. And you can kind of like shop, p-value shop by like different post hoc tests and different methods. So like one of them will give you 0.06 and then one of them will give you 0.045. And it's like, which one are you going to use? Seriously, like even if beforehand you were like, I'm going to use the one that ends up giving you a 0.06. I mean, how many people are not just going to, unless you have that 2-step thing where you tell the journal, here's the experiment I'm going to do, and here's the method, the statistical method I'm going to use. Now you're stuck, right? But if you you don't do that, which basically nobody does, then you can just go around and shop. It would look a little weird, like maybe if your lab group always uses one test and then you have this one experiment that for some reason you're like, hey, I felt like using this other one. And your p-value is like really close to 0.05. That'd be pretty suspicious, pretty obvious that you're probably using that different test to p-hack. So that's basically where what the real problem is what is it, type 1 errors, right? False positives.
I would even imagine type 2 errors would be more important, because if you have a false negative, let's see, false positive, you basically state that null hypothesis is rejected, and then false negative, you would state that it's not rejected. So yeah, it depends on the question more than.
But for p hacking specifically, what people are kind of trying to do is take.
Yes, with alpha.
Yeah, what they're trying to take is data that really does not convincingly, according to the conventions that are in use, does not convincingly support so-called rejecting the null hypothesis, but in just regular everyday scientist speech, the experiment having worked and produced significant results. So you shop around with these different methods to just try to, because, and again, and then it creates the problem, or the problem of, so publication bias for positive results is its own problem. It's its own problem, plus it undergirds all of these other problems, because all of the things that are not published, it's like this probabilistic underlayment of trials that are hidden from view.
Not the same sample space.
Right. So if you, like, for example, you know, I always thought it was funny ever since I was a little kid, skateboarders, now, like, you know, trigger warning if you're a skateboarder, but there's this thing they do where if they do a certain, like, what, I don't even know the terms anymore, ollie,
a kickflip,
fakey nose bone, like, like with some kind of like fancy kickflip, right? Or grinding, like some Tony Hawk shit. If they've done it once in their lives and they've busted their ass 1,000,000 times, they'll be like, I can do that. Like there's like, they'll say, I can do this move if they have done it once.
That was me.
Like what they really mean is I have done this once, but they'll just say like, oh, I can do that. Like I can do a such and such. And so that's an example of like sort of this, you know, all of the trials where they fell on their ass is like left out to the side. Because if you said, okay, here's like, what's your ratio, what's your odds ratio, like 1 in 500 tries or something, somebody might not take that to mean that you can do the thing. But if you just say, oh yeah, I can do it, I've done it, that sounds convincing. So you can imagine a scientific experiment, right? Like I want to see if this drug lowers blood glucose in this model, this animal model of type 2, you know, nutritionally induced or whatever diabetes. And you do a few trials and you feel like you didn't quite get the experiment right or whatever. Now, for an animal trial like that, it's pretty hard to just brush those results under the rug. What's really easy is molecular biology, because you just, you know, you get a plate, you do maybe a PCR or you do something, and
oh, I see what you're saying. Yes,
and then you go, oh, it didn't work, throw it out. And, you know, oh, it didn't work, throw it out. Oh, it didn't work, throw it out. And then you have one where you get this real sexy looking result where like the gene expression, the gene you wanted to be up, way up against control group, way up, everything else not changed, you know, which, you know, good, everything looks nice. And you're like the skateboarder, you're like this treatment does this result. And of course you don't just write up, I did this experiment, you know, I did 20 plate readings and I kept the one, or like if you do a gel with like a antibody, you know, a Western blot, and one of them looks awesome and
all the rest,
5 of them look like smears, you know, and one of them looks like this like bright neon bar. And then you put it in the paper and you say representative Western blot. So it's like the skateboarder, right?
I think with molecular biology specifically, it's such a big one, because I think it's one of the fields that I've read about where there's so much big data on mechanisms and single molecular interactions that are never repeated. And the reason could be I think it's two specific problems here. So the first one is obviously systematic error, where it's like you're just not doing the experiment correctly with the measurements, or the gel material is not exactly the right ratio, etc. So the experiment itself is going to fail. In that case, okay, that makes sense. You're just not doing things wrong until you get the method correctly. Then we analyze that information. But then it's like random error, where it's like, well, there's actually other influences here, and you're not taking account of that. You're just throwing out the gels that might have an antibody that can bind to both epitopes. And then it's like, well, you only chose one. You completely left out that data. Another molecular biologist tries to replicate this, and he sees completely inaccurate results. It's like, no wonder why. So I really do think it depends on the technician, how experienced they are, and obviously how much is really known about the system. It's like you don't know, like what you're saying. It's a research-based approach. You don't know what you're expecting.
Yeah, so a lot of molecular biology, because the techniques can be challenging, like you can spend a year trying to design an antibody that binds to what you want it to bind to, doesn't bind to other stuff reliably, right? So you're in this mind space where if something doesn't work, it's because it didn't work in the sense that most people use that term, like you did it wrong. It's not. But then you start to bleed into, is my experiment just wrong-headed? And there's, actually the antibody, if the experiment, the phenomenon I'm trying to measure was happening the way that I'm saying it's happening, this antibody would be fine and it would work. And I'm actually just chasing, I'm acting like I'm making a technical mistake. And it kind of, it can bleed together because, and then it gets to the question like, well, what does it mean for something to work? Like if you can do a surgical technique and fix somebody's trigeminal nerve and like not cause like desiccation that messes up their salivary glands, say, whatever. And you can do that, but like you have to be, Dr. Strange, like the best surgeon in the world or whatever, or like robotics. Or it's like a 1 in 1,000 that there's no damage or whatever. I mean, it's like, does that technique work? Like, cause like, because like you can't just give it to any surgeon. Like, say you have to be the top, X% of surgeons in the world to like do it and not cause the outcome. So it's like, does the technique not work if you have a negative outcome, or the skill of this? Because in the real world, things are, can be applied in ways that aren't just like, like abstractly just perfectly. And even in science, like I used to bring this up a lot, but like even just a glucometer, it's measuring the conductance, of blood with a glucose oxidizing enzyme that creates a current. So like, you could just like the chip in there could be reading like what you're. So it's because a lot of people, they look at a tool and they're like, just in their minds, maybe even doctors, they have this idea that there's like a little eye in there that's like counting glucose. And that's not, and that's like, 2 degrees of separation away, right? Because it's like the glucose interacts with the enzyme, releases electrons, that creates a current that's picked up by the actual machine. So it's like kind of a 2-step thing. But there's, experiments that have like, I don't even know how many steps, but way more than 2. So it's like, what are you even measuring? And each step along that path could have some type of failure, whether human or just, whatever, random. Actually, somebody asked a question. This is, does randomness exist? No DMT jokes. Thanks.
Randomness exists. I would definitely say it has to, from the entropic definition of randomness. It's like that is the reason why there's even such a thing as categorizing the forward motion of time. It's because of entropy, and things naturally become more random with time. And randomness in this case wouldn't be what we think about colloquially as things are random, like it's all over the place. Rather, it's the lowest energetic state possible for any system, and that it just happens that we categorize that as randomness.
Okay. Yeah, the way I usually think about, well, first of all, if randomness doesn't exist, that really doesn't affect what we should do, because generally speaking, I don't think we would ever have observational power enough to like hone in on things such that we no longer have a probabilistic view of things, but just like a purely causative. Mm-hmm. So effectively, we would be operating in probabilistic, whether things are deterministic or probabilistic. Now, in terms of like if they actually could be, I don't know. I guess I'm a little like Platonic
idealism.
Yeah, and I sort of have the Einstein, like, God doesn't throw dice. Like, I do sort of feel like,
No, I agree
yeah,
that there is some of that without a doubt. It's just the randomness that we attribute as it's sporadic can still be a very controlled setting. It's not like this isn't without reason. The randomness plays a role in everything, and in a sense, it's not without causality. I guess like the perfect way to think about it is electrons can be distributed
yeah,
in their
electrons.
cloud
let's
at
let's just go there.
like random intervals, but the cloud shape itself is important. It's like it's random within the clouds, but it's ordered within those random points. And that allows the reactions to occur in the way that we know it to occur every single time, a straight one-to-one correspondence. So, yeah, I agree. It's like it's just because things are random doesn't mean it's necessarily without order in a sense.
Yeah, well, so, but, to get to with the electron, you know, we can't really observe the single electron, in the sense that you can observe a car.
There's some physicists that argue. Never mind. I'm not even going to go there.
But okay,
Go
let's
on.
just say if we couldn't, and we could just say, okay, well, this is the electron cloud of the electron, the first electron of this orbital in this atom, okay, and because the thing about those orbitals is you can never from the probabilistic perspective, you can't say there's a 0% chance of it being anywhere, like even on the other side of the universe. There's just like, it's like a 6 sigma event that it would be over there.
It's a statistically significant event.
Yeah, it could Ryan Gosling itself over there.
Right.
It could, yeah. It's like, what do you do? Like, electron, like, why are you with this? Like, I drive. But that single electron, could there be, if you had some type of insane n-body problem accounting for every bit of matter and energy in, just like one little box, like an enclosed experimental system with some atoms and, air atoms or whatever, and you can observe those air atoms and exactly what the temperature, the energy going in, coming out, how many atoms are in there, and their movement, and you have like an equation to predict like the actual air atoms moving around, so like what's going on. Could you get to the point where you're like, we can just track this one electron, and like we know that it's going to go 10 minutes from now, it's going to be exactly in this point, this one?
I think they actually can. It does become like there's some methods to the deterministic positioning of subatomic particles, but then that plays into I should know this like immediately off the top of my head since I'm always surrounded by this. If you know the velocity, if you know the position, you can't know the velocity. What is that? The law?
Oh, the Heisenberg.
Heisenberg's uncertainty principle.
Yeah.
And so
that's kind of what I'm talking about.
Yeah.
So could we break that? Is basically, I just asked a long version of that question.
With the current mathematical models, from my current understanding, and I bet you that there's exceptions to this, you can't. But it's not impossible in the sense that maybe a new model that encompasses something else could make sense of the answer. And that's what I was going to say to you about like these physicists. There are physicists that believe there's such a thing as a single electron universe, where it's like the reason why there's a zero probability, there's never a zero probability that it's anywhere, is because it can be everywhere at once, and it's the same electron, just in different oscillations and different positions in space. It's crazy to think about. But yeah, it depends. I think it depends a lot on the math model that they use and the current limitations of how we perceive physics.
So I've never really gotten that deep into physics. I even considered doing physics, but I've never been like a math wunderkind.
Yeah, you should. I strongly recommend you would become that much better biologist, in my opinion. But yet the math, you're going to need a lot of math if you want to do like good physics.
Yeah. So, but is the reason for the Heisenberg uncertainty principle that to measure, so if you're looking at the velocity of an electron,
me pull it up.
you
Go.
get that, but now it's sort of like an n-body problem where you if you know, in order to measure its movement for its velocity, it has to be, you measure it where it was, it's in a past point. So now it's in a future point. So you can't know where it is. Whereas to pinpoint where it is, you have to look at it statically, which precludes knowing its movement velocity. I mean, is that the reason? Or because that's what I'm assuming is the reason why Heisenberg is uncertain.
No, it seems like it's different. It comes down to get the probability density rather than like what you're stating about if you know one event, then the other might be changed. It just seems like in general, because of the probabilistic nature, it changes what you can measure with certainty. So at any one event, it's the same thing.
Okay, so the method that they have, you have to pick one to get. You can't. You just can't get both. I don't know. Maybe that's just a better way of what I was saying. I don't know.
I'm gonna have to sit down and go through it.
So as we're speaking about this weird stuff, so we went over the history of the p-value and its accoutrement, and we went over, you know, sort of unweird, not weird, like not weird. Is there such a thing as probability kind of factors that are wrong with it, right? Like regular stuff, like it's, you know, it does this but not this, and it's used for this, and that's inappropriate or whatever. So now the weirdness. So we hinted at it with the, you know, you've got a drawer full of unpublished negative results, but there's this journal somewhere where there's the one positive result in the paper. So what's up with that? Does that affect the p-value? So the fact that you might have done the experiment more times than you're reporting, you know, because say you do the experiment a bunch of times, maybe that changes your statistical power. That changes what your, how much confidence you can have in your result, what you can report as a p-value, because you should really include all of those prior experiments, maybe. And is somebody keeping track of these probabilities? Like if you open up the Schrodinger box or whatever with the cat, not cat, you do it a bunch of times. Like, is somebody keeping track of how much cat, how much not cat? Which is colloquially referred to as the Monty Hall problem, off of some stupid TV show. I think I have the wiki. So I'm going to take full advantage of OBS features. And so we're done with the paper. I had the paper up for a while. I'm going to trash that and instead Monty Hall problem.
Really quick, just to go back to explaining the Heisenberg uncertainty principle. It's because as soon as you try to observe the other, you're doing it with photons, and the momenta of the photon is enough to cause a large enough deviation in the electron with either position or velocity, and so it's never the same result. Essentially, it's like when you throw a basketball, you're measuring the basketball's position and velocity. The things you're measuring it with don't have a large enough effect on it, and so it's just this discrete value. With electrons, because they're interacting with photons, it'll never be an 100% accurate value.
Okay.
So yeah, totally independent of what we both thought.
That kind of reminds me of, you know, that thing where if you're observing a photon, it collapses the waveform.
You're observing the photon, it collapses.
The eye slit, the double slit experiment.
Okay, yeah.
Yeah, so I've heard people say that that's not legit because they just use the term observe very neutrally, like, oh, we observe it. But what the observation actually is you're hitting it with something, or it's passing through something so that you can observe it, right? Because how do you observe a photon? Right? You have to interact with it. And so I've heard some people say that it is the nature of that interaction that is causing the waveform to collapse and for it to act like a particle rather than a wave. But I've also heard that there have been more elegant experiments that have asked that question more and gotten also weird results. So, I mean, I'm open for the weirdness.
This comes down to the interpretations of quantum physics. There are multiple different ways that, so there's like groups
Did
of
we
physicists.
get this far without saying quantum?
Yes, actually we did, which is nice because it's the same stuff. It's just quantized. That's all it means. But
It's such a buzzword. I'm really proud we made it like 2 hours without hitting it yet.
We're not saying it again.
No, we can say it. I just think it's funny.
Subatomic mechanics. Essentially, there's like groups of people, just like there are groups of biologists. Like we really think that bioelectricity plays a large role, but then there's people that think genetics plays a larger role. People that think secondary signals. In subatomic mechanics, there are people that believe a Copenhagen interpretation, where it's like the more colloquial sense of probabilities. There's people that believe a pilot wave theory. They call that Bohmian mechanics after the guy that created it, Bohm. And that is more deterministic. And then there's people that believe a more conscious aspect, where it's like your own consciousness affects the results. And the list goes on. So yeah, there's different interpretations for and by different groups. But the one that's academically supported is the Copenhagen interpretation. And the one that's like right behind it is the pilot wave theory or Bohmian mechanics.
Okay. You know, speaking of this weird physics, have you seen, there was a paper, I think, that just came out. Somebody shared it that I follow, like Keith Woods or somebody like that. It was the quantum entanglement paper.
The portal that they did in like Caltech or something?
I think so, yeah.
Yeah, they created like a mini black hole to entangle two particles through it. Two particles containing information. I think it was like qubits.
Yeah, and I think they were trying to ask the question of whether fundamentally particles are connected.
Yeah, all of them at the same time, right?
Yeah, just, you know, not to the extent, obviously, that they can't have discrete interactions, but there's, yeah, I guess I have like a pet theory that at the end of the rainbow on physics is probably just like Platonism.
Yes, I would agree. The Platonic ideal. In fact, when you even read, like when I go through my physics textbooks, all of their models are what they state for idealized systems. You make a smaller version that's more perfect, and you can ignore factors that might influence the result, get the same effect anyways. And it's like with Platonic idealism, there is an ideal world where things work in a very systematic, deterministic manner. I do think that at the end, we're going to wrap around right back to where we started with classical mechanics, where it's like everything is determined. If you know the position, the velocity, whatever, you can predict 100 years down the road what's going to happen with that thing you're looking at. Whereas in quantum right now, it's like it's more probabilistic, but it might return back to a deterministic nature. There's a lot of physicists that think this way.
Yes, return to tradition.
Return, yeah. Is the apple falling from the tree?
Yeah, I mean, I'm thinking like if you had, you know, one triangle, right? You could be like, okay, Platonic triangle. But then if you look around the world, unfortunately, you know, our eyes have a certain resolution, not so great. Resolving ability, I should say. Even our cameras have a certain resolving ability in terms of subatomic particles. Really nothing to, not good enough. But that doesn't mean that the world isn't, and I'm just saying triangles just because that's my favorite shape, but that doesn't mean that the universe isn't a fabric of n triangles that you just can't appreciate ideally because they overlap with each other and mess up your observation ability. But if you could account, you know, for the interactions of all these things sort of overlapping, that you'd be able to see that more clearly. So it might have just been a technical issue. Like we have these mechanics, and then the technical observation outstrips the mechanics ability to, you know, it starts looking dirty because now you're observing things that you're observing too much noise, but then not enough to get past the noise. It's like there's this zone of like, it's like, you know, a forest kind of thing. It's like, you know, you can see all the trees, and then there's a point where like you can't see it anymore, and then there's a point where you can see it again or whatever. I don't know, like it's useful. a useful zooming distance and like somewhere in between a non-useful zooming distance. I triangles. Somebody said I triangles. Yes. I is triangles. Okay, so real quick, the Monty Hall problem, because this is like the
We just jumped around. Yeah.
yeah.
Okay.
This to me is just this sort of poster child for how whether you're talking about statistics in this case, but math in general, physics, things like people they want to take something and maybe throw it out if it's not intuitive. But from what I understand, this phenomenon has been modeled, has like experimentally, and always replicates, so we have replication, right? Like the replication crisis. We actually didn't talk about that either. That's another buzzword. Replication crisis, particularly in the psychometrics. By the way, do you know what's the most replicable or one of the most replicable data sets in all of psychometrics? Care to guess? It's we could say, to be polite, certain groups that aren't real but maybe have like ignorant, unworldly people may categorize them as in a group based on surface level features, may categorize certain humans in certain groups, and that those different groups would score consistently different on IQ tests.
Physiognomy, essentially.
Well, basically, groups of humans from different places and IQ levels.
That's the most replicated data set.
Yeah, to the extent that they've been actively trying to not replicate it.
Huh.
And it's still, like, for example, the most simple one is, oh, well, the IQ test is culturally biased. Like certain people won't be as familiar with these words for reasons outside of intelligence. And then, so, reformulate the test, try to make, to remove that confounder, if it is a confounder. And the results were more not the way they wanted them to be in that particular case. And I'm sure there's other it's basically like it's politically incorrect. It's politically untrue that phenomenon can exist. So we're just looking for the way that it's not correct.
Right.
We'll find it someday.
That's skewed distribution with that.
Yeah, they are trying to smear the p-value all across the spectrum there, and it just loves the left side. It loves the zero. So the Monty Hall problem is, imagine that you're, on some kind of stupid game show or whatever. There's 3 boxes, 3 doors, 3 anything, and behind 2 of them is nothing or something shitty, and behind 1 of them is a prize. So everybody knows that if you have no information, right? All you're presented with is 3 possibilities. You have a 1 in 3 chance. So if you repeated that experiment a million times, you should expect something really close to 0.333 successful guesses. Now, if the guesser was to make their guess, so door A, B, C, so they guess door A, and then the host says, well, it's not door B, so do you want to change your guess? If the guessees are the doors, I guess, changes their guess to door C. Now, what you might expect is that wouldn't change the probability, because you could have guessed C before, you could have guessed B before, you could have guessed A before. It's all third, right? And now if they had told you before you guessed at all, they said, well, it's not B. So now it would be what, 50-50, right? Because now you're basically just guessing between 2 doors or 2 boxes. But if you change your guess to C after they just tell you it's not B, then the probability, and hold on, I don't want to fuck this up.
Yeah, Yes, I got it. Essentially, the ability goes higher. You have a higher chance of getting the correct answer if you choose to switch it after you get the information from the host.
Right. And the specific numbers are when the player first makes their choice, there is a 2/3 chance that in this case, they're saying car is the prize, is behind one of the doors not chosen. In other words, a 1/3 chance that it is behind the chosen door. This probability does not change after the host reveals a goat, so goat or car, behind one of the unchosen doors. When the host provides information about the 2 unchosen doors, revealing that one of them does not have the car behind it, the 2/3 chance of the car being behind one of the unchosen doors rests on the unchosen and unrevealed door, as opposed to the 1/3 chance of the car behind the door the contestant chose initially. So you go from the original 1/3 chance. So in other words, if you did this a million times and the contestant does not change their door, they will preserve the 1/3, the 0.333 long run success. If they change it, they go to
2/3.
2/3. So they double their chances.
Mm-hmm.
Even more than 50-50.
Yeah.
And apparently this was controversial. Supposedly 10,000 readers, including nearly 1,000 with PhDs, wrote to the magazine who published this problem in Parade magazine to say that the guy who said he solved this, so he's the one who explained why it changed, vos savant. Even when given explanations, simulations, and formal mathematical proofs, many people still do not accept that switching is the best strategy. And then it mentions the most prolific mathematician. He was not convinced until he was shown a computer simulation. So in other words, they've done, as I was saying, like if you repeated just picking 3, a computer simulation would you could do 1 trillion iterations. It'll give you 0.333, unless we're observing a 6 sigma event. Unless the computer is a sigma male computer.
I knew it.
But and
drive forever plane.
And then if you do this switcheroo after you're given the extra info after your initial choice, so what makes it weird is that it gives the impression that that God is up there with a bean counter keeping track of when you're giving the information. Because the door is real. The goats are real. The cars are real. The information feels just like this abstract, like it's like, either there is or there isn't a goat/car behind these goddamn doors. What's going on? But
right. I see where you...
So that kind of gets to why statistics can be important, because specifically, so again, imagine the stupid skateboarder who says he can do a trick that he can only do once out of 1,000 tries, or the molecular biology experiment that threw out a bunch of iterations that didn't work, but had one that did. It's like they're kind of showing you, they're acting like they got the car, but it's like, dude, you got goat. Like you opened a bunch of goat, and then you opened like one car, and you're hitting like less than 33% chance, and it's like that's not right. Like all those previous experiments, assuming they were done correctly the same way, count, and you should actually modify your statistical rigor calculations to account for that, because like the coin toss thing, it's like the universe really cares how many times you toss the coin.
Even if you do like a simple chi-squared model, actually not even, if you do like a simple probability commutative model where it's like how many times the experiment was performed times how many times it was performed correctly times how many times it was published, it's like even there, it's not giving the probability that you'd expect a majority of the time. It's the first event times the second event times the third event, which will give you a smaller number in general. So it's like, again, the niche result is what gets published. and the niche result is taken as a equal distribution, meaning like it's part of where whatever was taken from the population, the method population here, when it doesn't reflect that, that's like the best result that you could have possibly gotten. Essentially, you got the car and not the goat, and you're showing people that your results would always replicate with the car instead of the goat. When in reality, if you take all the information a priori, it's like no, you failed a couple of times, and you're misrepresenting this information to people through statistics.
Yes. Yeah. You're kind of like artificially. It's kind of like if you could pretend like you have a bigger sample size, because like if you have a bigger sample size, you get away with more power in your analysis. It's like if you could pretend you have a bigger sample size. This is like the reciprocal. It's like if you could pretend you had less iterations, it gives you more leeway.
Even with like sample size calculations, to get a good beta, it doesn't guarantee that your results are any more representative of an alternative hypothesis. It's like most people just, I want to say most people, medical research itself will not specify a predictive alternative hypothesis beforehand. And so it can be anything. And so it doesn't matter, like they love to say, well, my study was powered. Powered for what? What are you trying to prove? Does that make sense?
Yeah, power is. I actually don't. By the way, can you hear me? Because somebody said that my audio was left ear only, so I just tried to flipperoo something around. Hopefully it's stereo now, but who knows? I don't know. It's driving me crazy. Yeah, I know like power. I like my relationship with the power calculation is just like getting, having my PI be like, you need more animals. Because I think power is one of those things that like, would be sort of impossible to calculate by hand, but you just kind of, you give the parameters into a software. So yeah, exactly. But yeah, the power, and again, the power is just referring to the
false positive, false negative.
Right. The p-value, which again is a random assessment. It's a random value that is related to not your data and hypothetical random data, or the possibility of hypothetically random data that is your data or more extreme than your data. The left ear is louder. See, how could that happen?
I hear you perfectly. Maybe it's like they have headphones on.
I'm not, but no, it's two people are saying this, that my audio is left ear.
Oh man,
that's like the weirdest thing.
You over prepare too. You're always making sure everything works, and there's always some little issue that comes up.
I guess that's not the worst thing. I mean, as long as it's I guess I'll just go. I mean, if I go back and listen to it and it sounds really garbage, I'm going to be quite sad. But
honestly, like the most important thing I've got from this paper that you sent, and just looking at this again, is truly you cannot state causality if you go with a frequentist approach, p-value, significance testing, null-hypothesis testing, whatever which manner. And that's the thing that most of medical research is looking for. They want the randomized controlled trial. Even then, like the words randomization, what is that for? That's to reduce selection bias. Controlled, whatever you think might be affecting the results, another confounder. And then blinding to prevent like a positive outcome when there really doesn't exist one. So you're reducing type 2 and type 1 errors. But none of that even matters in the scale of things, because it's still not possible to denote causality with these methodological research tools. And it's promoted as such, that if you do a randomized controlled trial and you control for confounders and you get a statistically significant result, that means you've determined causality. And we know that's not the case, because a lot of things and a lot of information has changed since a lot of these prior outcomes were measured.
Right. Yeah. They're still commenting on the audio.
Too, or?
It's just my audio. Okay. Yeah. and even the idea of, I think just going down the road of like when you're sitting there and you're trying to achieve, if you're ever doing science and you're having a problem where you're like, I have this data, I want to publish it, and I'm just trying to get p below 0.05, you really got to ask yourself what you're doing at that point.
Your life.
Because what we really want, I mean, what I would want, I mean, I wouldn't expect, or I wouldn't say that an experiment is successful if I could just kind of show some small, statistically significantly different amount of like say that, you ablate the beta cells, and then, 4 or 5 regenerate without any treatment. And then you've got a treatment and you've got 6, and then 5, and then, and you've got, and it's like consistently more. So you're like, hey, this is statistically, because that could happen, right? You could just have a small effect, but if it's very consistent over a large N, then, you could pretty easily get below the alpha of 0.05 with that for your p-value. Not that hard. But what the fuck does that matter?
Yeah.
Because 6 beta cells isn't enough to maintain glycemia any more than 5. in a zebrafish. I guess in a human, the numbers would be different, but same thing. So, yeah, it's like what you really want is something that sort of can eventually translate into like a rather obvious relationship. Like, enzymes work slower when they're cold. Like we don't really need statistics for that. Like we might need statistics to be like, okay, this enzyme from this organism, this is how much it's affected by temperature, because they're all different. But like, there's just this sort of underlying law of that relationship. Like there's different ways to look at it, and we can kind of accept that and make sense in a bunch of different ways. And so that's the kind of stuff that I would prefer. But yeah, statistics. I guess maybe the counter people would just say, well, we need something. We need some. We're trying to look at things that might have small effects because we're trying to like, maybe this drug has this effect, but then if we can show that it has a significant effect, we could modify the drug, do another experiment. Maybe it'll be higher or whatever.
Their working heuristic is at least we get it right 80% of the time, essentially. That's the heuristic here. It's like, well, we have this tool and it seems to work a majority of the time. So even with the talking points that we've mentioned, still, if they grant all of that correct, 100%, everything that we've stated is fact, then 80% of the time, their methods still work. And that would be their defense, essentially. Like, they're still successful. And to that, I would say your methods would be successful 80% of the time as a passing grade to you. But to me, you would want that to be closer to 100% of the time, as great possible success rate as you can get. So I think it depends on the philosophy. We are looking for deterministic outcomes because determinism means we have predictive power. Saying forget the determinism because it's very hard to get that predictive power in medicine. Just do the best that you can.
Right. Yeah. I mean, speaking of determinism, if I saw, for example, like I think in one of Michael Levin's papers, he had the, this, whatever the treatment was, like 50% of the time makes a ectopic eye or whatever. So the one philosophy, maybe for people that are into like p-values and that kind of science, would be, well, it's a 50% likely event. But what I would say is, well, it's probably 100% likely if you can figure out, like, there's probably something about the,
yes, I see what you're saying.
The frogs, like whether it's their genotype or something about their treatment, their this, that, the other thing, where like whatever's actually the thing causing the phenomenon to happen, it's not that the treatment causes the phenomenon. Or in the sense, like, you know, like if you take testosterone, right? Like testosterone doesn't make your muscles big. Like it goes, you know, it goes into the cell and it tells muscle cells to synthesize
Change.
protein, you know, muscle fiber.
Mm-hmm.
If you could like use, you know, electromagnetism, radio waves or whatever to like hit the body and aim it, you know, at the DNA and like tell muscle cells to do that, it would have the same effect. So it's like, what's the, you know, but of course we understand like when we say cause for certain things, but other people, you know, when it becomes these like more research questions, when they think of cause, they think of like a 1-to-1, like a no degrees of separation, where it's like, I would say probably, you know, the probabilistic results that people get are because, yeah, they're, and you could say it is true, like in a population, like this drug is going to have this effect. You can expect it in like 70% of the people and 30% are going to be non-responders. But what I think that is 70% of the population is a subgroup called responders, for which 100% of the time it works, and 30%, you know, and
that's a good way to look at it.
I would be very confident that if you could really, really look into those people, you would find the reason, you know.
This is where it plays into Bayesian statistics again. Is you're looking for a priori knowledge to build upon to get the likelihood ratio as close as possible to the outcome you're looking for. So the drug needs to work. We need to identify the mechanisms. By identifying the mechanisms, we build our repertoire to get a more accurate result, a higher percentage of the time that we investigate this, which is the right approach with medical research instead of the frequentist approach, where it's like you do enough, you'll get a set of data, and then you kind of see what you can say about the data. 95% of the time we get the result we want, 5% we don't. Again, I think the philosophy plays a role here, and I agree with you 100% with your line of thinking and the results that would come from that line of thinking.
Yeah. So, yeah, actually, there's another thing about big data actually that I was thinking about, because that's kind of how big data works. Is that, you know, people will say, oh, you know, look at these bigger is always better, and population-wide stuff. But if you looked at, for example, I don't even know if I used this. I may have used this example before, like in a video, but a rare disease, a rare genetic condition like leptin deficiency, just straight up leptin deficiency. It's like very rare, and these people will just eat food forever, right? They'll just never stop unless they get leptin treatments. The incidence is so rare that if you tried to look at it the way that people look at population level stuff for like drug effects, it would be zero, or it would be, you know, you would be able to confidently accept the null hypothesis that leptin deficiency does not exist in the population, because the amount of hits of leptin deficiency would be, you know, you would end up with, if you were trying to show that there are some people with leptin, you know what I'm saying? Like,
yes, like the null hypothesis would be there are no people with leptin deficiency, and then you find these people that is so rare. It's like you reject the null hypothesis because clearly they exist, and it's very small percentage alpha, essentially.
Well, yeah, you'd have to like, you basically couldn't do it the way that people normally do it. But of course, you know, nobody would take that seriously, because all you really have to do is show that there's one person with leptin deficiency. And the way you show that they have leptin deficiency isn't statistical. You don't take a bunch of people. It's the same way, like, you know, people have 5 fingers. Like, you don't take the average fingers of everybody and then be like, people have 5 fingers. You can just kind of like, well, I mean, you could do that, because sometimes there's somebody with 6 fingers. But it would be negligible. But to say like people in general, you know, have 5 fingers. So what you have to do is show that somebody, you know, do their genotype or whatever, or show that like giving them leptin drastically changes their appetitive drive, stuff like that. It doesn't matter if there's only one in the world. You know, that would be statistically insignificant according to like how a lot of, drugs or dietary stuff is experimented on. So what I'm saying is a lot of things that are considered like, how like every year, I don't know if they're doing this anymore, but I remember ever since I was a kid through like my 20s and stuff, every other year it felt like, oh, eggs are bad for you. Express eggs are bad for you. Eggs not bad for you, bad for you. But, or like fiber, like, oh, giving old people the bran muffin thing, like, oh, it decreases colorectal cancer, doesn't decrease colorectal cancer, doesn't. And it's like, don't you think it's a little more likely that there's some subset of people for whom that fiber in their diet, is going to like prevent colorectal cancer, or at least like slow down the progression or whatever the hell causes them to be in the group that's, ends up not having colorectal cancer, and that other people get colorectal cancer in a different way, or they process fiber in a different way, or whatever the hell. And the same with eggs, right? Like we know that there's people for whom dietary cholesterol doesn't affect their blood cholesterol, and some who does, and then what that cholesterol then does in people's blood is different person to person and all kinds of stuff. So like, but you still have like, it still ends up on the news where it's like, hey, the guy reading the teleprompter, he's like, experts, a new study shows that actually, eating 6 eggs a week has no negative effect on cardiac outcomes. And then like the next year, it's the same thing. And it's like people are just chasing, like with this big data, it's like, you could just change the question you're asking. Like, stop just throwing math at it and like repeat, because you could show all kinds of stuff. Obviously, every other year, eggs don't become disease causing and not disease causing. That's really unlikely. That's a Six Sigma event. That's like every other year, eggs have a negative effect or not a negative effect. Yeah. I think we could just throw that one out. So, if the result of the research is that every other year eggs seem to produce in the experiments a negative effect or not, then I think there's a problem with that, the way that research is being conducted.
That's the observational models. And once again, it's like they try to draw correlations to test with a randomized controlled trial that they'll never do. They're never going to take eggs and just give eggs in a metabolic ward to people to causality. And even if they did, none of those methods would still denote causalities. Like you're chasing a dream that does, like you're chasing the idealized version of causal relations that just don't exist.
Right. And you're not parsing out people. Like maybe maybe there are people that, whether it's because of other things in their diet or their genetic backgrounds or what the hell ever, are negatively affected by eggs, and others that are not. And if that's true, then I mean, let's just call that the alternative hypothesis. And the null hypothesis would be eggs affect everybody the same.
Okay.
So then our prediction would be, if the null hypothesis was true, that eggs affect everybody the same, there would be a very low likelihood of having alternating experimental results from egg trials. Whereas the alternative hypothesis would predict that there would be, this one shows negative effects, this one doesn't. This one shows negative effects, this one doesn't.
Those would be two different alternative hypotheses too. It's like you have to do the test in a different way for both. Right.
Well, I'm just saying, like, even like it's sort of like who watches the watchers, like who like assesses the actual, like the research that is coming up with statistics and a p-value. How about we take their research and make an hypothesis that it's garbage, and and predict like and be like, okay, well, if it was garbage, then you would expect the results to be like evenly distributed across positive and negative results for this experiment, which it seems to be. Like if you're basically getting a random distribution of eggs are good or not, or bran muffins for old people are good or not, then you're in that you're in that null hypothesis zone where your experiments are not looking at anything real. because if you were looking at something real, long run experiments would skew it towards, you know, having a consistent result one way or the other. Not that it would always be the same, but like, yeah. I probably had other notes, but
Gone through them.
I'm kind of fried.
I think I got through most of the stuff.
Somebody asked about Vonderplanet's raw meat. I did a whole stream on that with Leo.
Yeah, you've done it for a long time, actually, from what I remember.
Yeah, I've talked about him several times. I mean, my assessment is that he made up the events that supposedly happened in his book. I don't think happened. And I don't think that his model of how the human body works is correct.
Mm-hmm.
I don't think his model of what viruses and bacteria are or how they work is correct. I think raw meat's probably fine, probabilistically. I certainly wouldn't eat. I mean, if somebody just showed me like, hey, let's Agenus Vonderplanets this meat. There's a parasite in it. Look here, it's right here. I'm just not gonna.
What a way to put it. Oh, that's funny.
Like I do think parasites are real. Because see, I'm a biologist. I'm a biologist first. I really believe in biology, and that's a very profound lens to look at the world through. I believe it's true that there can be animals or microbes who are not just beneficial to us, that they have their own agenda to survive and reproduce, and that they could use our digestive tract, or in some really macabre and Lovecraftian, disgusting situations, even our brains, if they can get in there, fungal spores or worms or whatever. I've seen horrific medical imagery. And that they're not there to help us and to clean out dead cells. Maybe we have evolved. I mean, obviously, it seems very likely that we have evolved a commensal relationship with bacteria, certain bacteria more than others, in the gut, on the skin, any kind of orifice. And yeah, after a while, you get to the point where maybe evolutionarily, you would actually be worse off without some of them. So like you can have that kind of a thing. But then to take that idea and say everything in the world other than like man-made chemicals is there to help us. So like, don't be afraid of bacteria, like in high meat, you know, it's a detox. You're just detoxing. You're actually not having an infection. This is a detox because you have like industrial like toxins in your cells that specifically the bacteria in the high meat are getting rid of for you. They're helping you. I don't believe that's biological. To me, that's not a biological theory. It's like a natural hygiene Christianity sort of. It's like a, you know,
I see what.
And his book, that's what I said when I was with Leo. It's like his book basically reads like a sort of messiah figure. Like he's in the woods. He tries to sort of kill himself, starve himself to death. Wolves bring him meat, like wild wolves. I mean, that's his story. So that's what I think about Fire Planets. I met the guy though. I always say this because I don't think many people have. Obviously he's been dead for many years, but I've been doing this stuff for a long time. And I went to a lecture of his in Cherry Hill, New Jersey in 2006 or 7. And I mean, he looked pretty good he might have been like 60 at the time. He was a nice guy. I spoke to him a little bit. He gave a lecture that was weird. He said, he pointed to me as an example of jaundice. He was like, sometimes people that are too thin, because at the time I was just switching from like raw vegan to eating raw animal products mostly. And so, you know, the whole time I was doing all of the kind of weird diets, I was like too skinny. Even with a lot of meat, you know, because I wasn't eating a lot of carbs. And so it was kind of that kind of thing. So he was like, yeah, you know, if you don't, I forget exactly what he said, but I remember he specifically was like, yeah, people that are really thin, sometimes they have a little bit of like, you can see in their face, like jaundice symptoms. And he sort of pointed to me. So,
Weird, man.
yeah. That's not the reason that I don't believe in his diet, by the way. I think I actually am very honored that Agenus Vonderplanitz called me jaundiced. So yeah, any closing thoughts or
I guess really quickly on what you just mentioned about like pathogens versus commensal microbes. I've gone through this so many times with the people that deny viruses exist. They deny bacteria are bad. They deny anything is bad. It's pure terrain. And I've noticed a couple of things. One, they completely ignore any other evidence that suggests otherwise and focus and hone in on just the evidence that shows their points. That's itself a bias. And then two, when it comes to like bioremediation of bacteria, which is their talking point, it's like, well, you see bacteria breaking down things in environmental places for bioremediation. They don't mention what exactly they're doing, like what kind of enzyme, which kind of reaction, how effective is this? Is it a mass scale? They just kind of extrapolate that to, oh, it happens in the body when it's one, not usually the same microbe, doesn't have the same enzyme, doesn't catalyze the same reaction. It's like you can't just take this and apply it to humans because it doesn't exist. And then finally, the third is no alternative hypothesis, funny enough, for what causes diseases in the first place, aside from the environmental chemicals that they never really go in depth as to how it causes the disease. Does mercury cause every condition known to human beings? It's highly unlikely.
yeah, or yeah, like industrial revolution, soot and chemicals and stuff, and that was the origin of disease or whatever.
It's no doubt a huge factor. It's like you have to specify how, because otherwise you take a broad stroke with a brush and say everything is caused by this. You lose any meaning. It's just a statement. That's it.
Yeah, the whole terrain theory thing. That's again, it's like what I was saying about the bioelectricity. Like people have this, the light switch brain or whatever. Like if there's kind of some questions about one interpretation, then they just want an answer that is totally diametrically opposed to that. They can't imagine that there's sort of a combination of things. Like for example, if you're walking through the woods, like say you're an animal, I guess a human could work, maybe, I don't know. Say like a big, healthy looking animal, big, and there's wolves. They might just pass you on by. They're breezing you. Whereas if you look, you don't look so good, you're small, bite-sized, if you will. They set upon you as they see you, and they each, and so you might say like, oh, well, the wolves don't eat animals. They eat, it's the terrain of the animal. Like if the animal was healthy, the wolves wouldn't eat it. But it's like, is that really what's going on? Or is it just the wolves are making their own calculation? Like they've got their own thing going on. They're a biological entity. They have calculations. Like if they see a bear, there's 5 of them. Maybe they could take down a bear. I don't know how that shit works. But if they see like a bear cub, right, they can definitely take down a bear cub with like a very small chance of losing any of their members.
Very small alpha.
Yeah. You know what else I didn't say the whole time that I've kept thinking, like laughing to myself before we started the stream, that as soon as one of us said beta, I'd be like, beta, like Jesse Lee Peterson.
They're going to have to come up with a sigma now.
But yeah, so that's how I see, why can't bacteria just be that way? Like they sort of have, and of course they're not like animals. They don't have eyes. They don't see that you're healthy or sick or whatever. They don't see these things, but like they have
remote sensors.
Yeah. They could, and, or if you don't have the ability to like wash them out with bile or different secretions and stuff, they set up shop. It's not that they want to be your buddy and not set up shop because you have like really nice secretions and cleansing mechanisms in your gut. It's just, it's like you can't, like the mugger goes after the easy target, and all that kind of stuff. So I just on that surface level, like what you're saying, I just don't see like why somebody has to go full terrain, like as if that's like that disproves the idea of infectious agents. It doesn't. It's like yeah, everything has, you know, like if there's people that can be hit by a car and survive, and like that same blunt force impact because the person is like smaller or weaker or whatever will kill them. That has nothing to do with like whether cars hit, you know, the fact that there's disparate outcomes based on like your underlying health level or the underlying strength of your biology. That really doesn't disprove that the thing in question is like harmful per se.
Right. Yeah, I agree.
So there's
Yeah.
no questions. Evan, who is the only person to ever send a super chat, said that he made it. Although I don't even think I ever, like, I think you have to have a certain amount of money in your YouTube. I don't know. The whole thing with Google and YouTube, like, I haven't quite figured it out yet. But, you know, we're not doing it for money. We're doing it because
We're Sigmas.
as Redditors, as Sigma Grindset Reddit males, we hecking love to do a science. To be fair, you have to have a relatively high IQ to appreciate a heckin' done science that we like over here.
On P values. Oh my God.
Yeah.
Yeah, this is a super niche topic. Nobody wants to hear this stuff. They want to hear like, what does the B vitamin do to the cell for diabetes or something? I don't know. How to grow brain. That's it.
Now, see, so we're in the point of the show now where we're making jokes. So you have to say something like, and we're specifically making Reddit I fucking love science jokes. So like, you know, imagine like Rick and Morty, like, Morty, I turned myself into an alpha of less than 0.05, Morty. I'm p-value Rick.
The problem here is I never watched the show.
No, you don't watch the show. You see the memes that make fun of the people that watch the show.
Gotcha. Like Berserk, you don't read the manga. You just look at the pictures and extrapolate from there. Make your own story.
And you're still sad, even if you don't know the story. Just seeing sad guts is still sad. So, somebody just asked in the closing here, if you were to discuss statistical significance as a social construct, what would you say?
It's a social construct.
Well, it is a social construct. Yeah, I mean, it is truly arbitrary. I mean, the one way it's not arbitrary is that it's not random. So those are two different terms. Like an alpha of 0.05 is not a random choice, because a random choice we could imagine would be anything between 0 and 1. And 0.05 is much closer to 0 than it is to 1. So it's not a random choice, but it is an arbitrary choice, because I seriously doubt any reasonable person, even the scientists that use p-values for the majority of their publications, would, you know, if you ask them privately, say that results for which the p-value is 0.051 are not valid, and results for which the p-value is 0.049 are valid all of a sudden, right? Nobody actually thinks that. So in that sense, if that's not true, because if it was not arbitrary, if that was an actual like over under that is meaningful, so in that sense it is a social construct, as opposed to other natural phenomenon that are not social constructs but are rather natural constructs for which society can be a little confused.
I just thought about something with what you mentioned. 0.05 is closer to 0 than it is to 1. And then the math from Calculus 2 came in my mind of convergence and divergence, where it's like you can have an infinitely small number or an infinitely large number. It's like, wait a second, you can't really say that 0.05 is closer to 0. In fact, the null-hypothesis might actually be still accurate.
Well, that's discrete math in regular guy math.
Yeah.
0.95 is a higher, it's a higher number than
0.05. Right, I get
it. Okay, so thanks everybody for. I think we had on average, I was glancing a couple of times, I think we had an average of about 10 concurrent viewers, and it says that we've had about 110 views. So in other words, over the course of the stream, those 10 viewers were changed out approximately 10 times, or at least some of them, or maybe 11 times.
Oh,
So we've
man.
had a turnover of, and we've been streaming for 2 hours and 45 minutes, which means a turnover of approximately, let's see.
There goes my math. I'm done.
But anyway, yeah, so if anybody cares, because I do get messages from time to time, hey, when are you going to stream again, blah, 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 blah. So probably once a month is a nice, yeah, 120 divided by 10. That's pretty easy. So 12. But per hour. So divide that by 2 hours and 45 minutes, and we can get the turnover. Somebody's like doing the math about the viewer turnover. It doesn't matter.
Right.
Yeah. So, but yeah, we should do another. If you have, another paper, maybe in a few months or whatever that you want to discuss in such a fashion.
Statistics wise, or like anything?
Anything. Probably not statistics, actually.
Yeah. Like the most frustrating one.
But yeah, so if anybody wants to read this paper, I don't know if this is one of those papers where you need to have like a university affiliation to download, but I would just like to say, as always, for my audience, if that's true of a paper that I mention, you can just DM me and I can send it to you, because I do have that, and I don't want to bogart papers. What paper? The paper is called "When Null-Hypothesis Significance Testing is Unsuitable for Research: A Reassessment," published in 2017.
I'll post it on the chat.
In Frontiers in Human Neuroscience. Oh, okay. So I guess he's specifically targeting this to the neuroscience population, which is indicated by the fact that it's published in a neuroscience journal.
Mm.
All right. Thanks everybody for spending the old Sunday late morning, early afternoon
Friends.
with us. Yeah, it was a friendly time as always, and yeah, hit that like button, subscribe, ring a ding ding that bell, and thank you so much. Peace out.